Okay, we are now live in YouTube, so good morning for everyone there. We will start in about 5 minutes, I think. I don't see my clock. In about 8 minutes. Ay, tengo que apagarme. Ahí se escucha bien. Ajá. Sí, lo ponemos acá. ¿Posteas el link a todo el mundo? En Discord y en el SBE para que yo lo pueda transmitir. Nico, ¿puedes poner la pantalla como de presentación?
¿La pueden ver? ¿La
Sí,
puedes
sí,
ver?
sí.
Ok, perfecto.
Sí, la veo.
Perfecto.
We welcome everyone here on Zoom and on YouTube. We will going to start in about 5 minutes, so keep online, and we are ready to start in a few moments. Thank you. Okay, we will start in about 2 minutes. Thanks again for joining us here in Zoom. Also the people that hear us in YouTube. We are very happy to have you here, and we hope you enjoy this. Remember, if you have some questions, you can make it using the question and answer option in YouTube, I think in Zoom, or you can post some questions in the YouTube chat. We will check it to ask our presenters. As there are so many posters on biogeography session, we will enlarge the session, I think, to 14 to 2 p.m. to 3 p.m. UTC -5. So all the people that want to see the Discord, will join us in Discord to see the posters, can have more time. We have about 28 posters, so we hope you enjoy them, and you can use it Discord to look a lot of wonderful works. Okay, Nico, I think that we are on time, so we can start now.
Okay, perfect, Salva. Thank you. Okay, everybody can see that I moved the slide of the presentation, just to make sure. Yes? Okay, yes. Thank you. Okay, welcome to everyone to the Symposium of Biogeography in the Neotropics of the SBE meeting. Before to start our presentation, I just want to read the code of conduct that is very important during these online meetings. So first, turn off the microphones and cameras throughout the presentation. The only people allowed to speak are the hosts, moderators, and exhibitors. Assistants are allowed to speak during the allotted question time at the end of each presentation. Respect the request of those express their disagreement of being photographed, filmed, named, quoted, among other possible actions. Do not publishing others' private information, such as physical or email address, without their explicit permission. Exhibitors must review that the material they present in any type of participation is not offensive. The debate must be conducted in a formal tone and language, and no time should deliberate intimidation, ad hominem attacks, or trolling be carried out. The chat of presentation platform Zoom must be used respectfully. Interactions throughout this chat must be kept to a minimum to facilitate the management of questions and the organization of each symposium. So I want to welcome our first speaker, and she is Camila Ribas from Instituto Nacional de Pesquisas de Amazonia, Manaus, in Brazil and also from the American Museum of Natural History in New York. Her presentation title is Bird Diversification and Evolution of Amazonian Landscapes. You are very welcome, Camila. You can start now.
Thank you very much. Thanks for the invitation. I'm very happy always about talking about biogeography. So let me share here my presentation. Can you see it okay?
Perfect.
Okay. So I work in Manaus in Amazonia. I've been here for more than 10 years now. My main interest is biogeography, mainly the biogeography of Amazonia. And I work with birds, mostly because birds are a very good group to study biogeography, mainly in a place that is largely unknown as Amazonia. Bird taxonomy and distribution is a little better known, so it's a good model to study biogeography. What we know about species richness of birds in the world is that the Neotropics stands out as a place where there are many, many species. When we look inside the Neotropics, we can also see that most diversity, species richness, is located in northern South America, northern Andes, and lowland Amazonia. But when we look to Amazonia as a large biome in northern South America, it looks like a very large and green area, so far at least. But actually, Amazonia is a very heterogeneous region, and its heterogeneity is much influenced by the rivers, the river system. We can barely sort out Amazonian environments by environments that are seasonally flooded by the rivers and environments that are never flooded. So the seasonally flooded environments, Várzea and Igapó, have a biota that has evolved differently from the non-flooded terra firme forest biota. Also in Amazonia, there are patches of open vegetation, like white sand forests and savannas, that have this patchy distribution, but also contribute with a unique biota and a lot of diversity. For birds, the birds from the uplands, from the terra firme, are the best studied ones. Many people have been looking at these birds and at their endemism patterns for a while now. And there are more than 1,300 bird species in Amazonia, most of them in the uplands. And several authors have proposed that these birds are not homogeneously distributed in Amazonia, and they are distributed in areas of endemism. At least some of them have congruent distributions restricted to different areas of endemism. This means that many birds occur only like here and are not present in the rest of Amazonia. One important thing to notice is that most areas of endemism are delimited by the large Amazonian rivers, showing again the influence of the drainage system in this large region. Looking at areas of endemism for upland forest birds is important for biogeography, but also for conservation. If we look at what's happening in Amazonia now, we see that some areas of endemism, mostly from southeastern Amazonia, are almost gone. So there are threatened species that may be thought of as Amazonian species, but actually they are endemic to regions that are almost completely deforested. So when we look at patterns of endemism and patterns of species richness, we are looking at patterns, at things that we can see today regarding Amazonian diversity. But when we try to understand how these patterns came to be, we have to think about the historical processes that have led to this distribution of diversity. The most commonly discussed processes for generating Amazonian diversity are the Pleistocene refugia, which was proposed by Hafer in 1969 and is still broadly discussed. And what he proposed was that Amazonia went through cycles of expansion and retraction depending on climate change during the Pleistocene. Pleistocene is the last 2.6 million years ago. And during this time, the whole Earth went through cycles of colder and drier climate. And when the climate was cold and dry, Amazonia could have been fragmented. That's what Hafer proposed. And in this case, the forest would have been replaced by savannas. This is very discussed and we'll talk more about it. Another possibility, considering that areas of endemism are mainly delimited by large rivers, is that the evolution of drainage in Amazonia was responsible for diversification. The most accepted model for drainage evolution dates most changes to the Miocene. So it's common to see in the literature a dichotomy between Pleistocene refugia hypothesis and drainage and riverine barrier hypothesis. Pleistocene, so refugia would influence diversification in the Pleistocene, drainage evolution in the Miocene. And this is an easy dichotomy, but I'm gonna argue that it's too simplistic. Another option would be that actually diversification occurred in spite of landscape evolution, that the landscape maybe was not changing when species were diversifying, and current patterns of diversity are the result of dispersal over a stable landscape. So how do we address these questions? I think the first main question is if current diversity originated through differential dispersal over a stable landscape, or through diversification was driven by a dynamic landscape. So we need to understand the landscape history in order to discuss diversification. Also, we need to understand the timing and spatial pattern of species diversification for the different groups we're studying. And we need to understand the relationships of these birds we are studying with current environmental heterogeneity. So there are species that will be more typical, that will be exclusive, associated to flooded environments, species from upland forests, species from open environments. And we need to consider this habitat specificity when trying to understand biogeographic patterns. So the first thing I'll talk about is a little bit about Amazonian landscape history. Many ornithologists working with phylogeny and phylogeography of Amazonian birds agree that most species arose during the Quaternary, or at least in the last 3 million years. So what was happening in Amazonia during the Quaternary? The first debate comes to the glacial cycles. So Hafer was right, and Amazonia was replaced by savannas in the last glacial. The last glacial maximum happened at about 20,000 years ago. Or maybe he was not right and the forest was never replaced by savanna during glacial cycles. Recent evidence from Amazonia is sparse, but these two are the most important papers. They are based on records from speleothems from Amazonian caves. This one was the first one published. It was based on records from a cave in Peru. And recently, this other one, 2017, was published in Nature, describing a speleothem from a cave in Itaituba, Eastern Amazonia, Pará. And what they show is that the pattern is more complex than we thought. So what happens is that Eastern Amazonia has a climatic pattern different from Western Amazonia. And in the last glacial maximum, although Eastern Amazonia was actually drier than today, Western Amazonia was never very dry. So the variation in climate in the past in Amazonia is not unique of the whole basin. It seems to be different in eastern and western portions of the basin. And for the eastern portion of the basin, it's quite possible that there was a change in vegetation cover, because this means this change here means a reduction of 50% in moisture. So this should have caused changes in the vegetation. Also, during the Quaternary, we have recently shown that drainage evolution was still affecting the distribution of Amazonian habitats. These are a paper published in 2019 as well. It shows that changes in river discharge and the evolution of floodplain was induced by climate, precipitation change. So changes in precipitation also alter the sediment flow of floodplains in Western Amazonia. And this means that portions of floodplains were recently abandoned and became terra firme, upland forest, in the last 45,000 years ago, very recently. So there is we found evidence of a recent establishment of upland forest in central Amazonia, showing that the different Amazonian habitats are on an ongoing process of adjustment that is still linked to how the rivers change through time. So now, after showing that the landscape has been pretty much dynamic during the Quaternary, we can look at the birds and from the different environments and see what they can tell us. So let's start talking a little bit about birds from the upland forests that are the most studied and the ones we know better. After many years of work, in 2019, we published this study bringing together results from many students that have worked at INPA and at the Guilde Museum for the last, I don't know, 10, 15 years. And we joined results on 23 clades of upland forest birds, more than 1,000 individuals for which we have genetic data. We reconstructed for these birds the lineage delimitation and their phylogenetic relationships. And we found that most diversification events on these 23 clades happened in the last 3 million years. So this is the number of diversification events through time. And also that large rivers coincide with 75% of the genetic breaks among the lineages that we found in Amazonia. So there are some commonalities in time, recent diversification, and in rivers coinciding with splits, with genetic splits. But there is a lot of change in the order of splits related to the different riverine barriers. Also, we have seen that when we modeled climatic stability for these taxa in the last glacial maximum, we see that climate stability coincides with the presence of the oldest lineages that we found in the base of the Andes and in the Guiana Shield. And also we find signal of recent population expansion in most upland forest species that we studied. Again, pointing to a dynamic landscape in the recent past. This is a summary phylogeny joining the commonalities of the 23 phylogenies we built. We see that the general pattern is that the oldest diversification event is related to the Amazonas Solimões River. Then the next event related to the Negro, to the current position of the Negro, and the next oldest event related to the current position of the Madeira. We also optimized the ancestral areas for the different nodes for all the groups studied. And we see that ancestral distributions were mostly in the Napo, base of the Andes region, in the Guiana Shield, and in the Brazilian Shield, mostly western edge of the Brazilian Shield. And then splits that generated species from the southeastern Amazonian groups are more recent. So we propose that ancestral distributions of these birds were centered at the shields and the base of the Andes, and that climatic variation caused differential isolation and local extinctions, explaining these recent lineages that we find in southeastern Amazonia. So what we see is that during the Quaternary, we have both the effect of rivers as barriers and climate on the diversification of upland bird species. So the rivers, they act as temporally dynamic barriers with varying degrees of permeability through time. And the dispersal limitation will depend on the ecological characteristics of taxa. So the pattern is quite complex. Then we can look at timing and spatial pattern of diversification for birds associated to the flooded biomes, Várzea and Igapó. This has started more recently. We still know very little about these birds that only occur in the flooded forests. But it became a very important issue, both because we know these birds and this biota are very little, and also because they are quite endangered as well. So the Amazon wetlands cover 14% of the Amazon basin, about 840,000 square kilometers, and they are recently quite threatened by the Amazonian countries' project of building dams in several large Amazonian rivers. The first study we did, we published, we are working on many others, but the first published study was in 2018. It studied phylogeography of what was considered one single species of bird from the riverine islands throughout Amazonia. And we used mitochondrial DNA and UC loci. And what we found was that there are at least 4 different lineages within this so-called one species. And we found a very recent history of isolation and secondary contact among these lineages. So we see the first split separating western from eastern Amazonia. And in eastern Amazonia, a lot of changes in population sizes and in isolation and contact, with hybridization happening between some eastern lineages, everything happening in a very recent time frame. When we studied some other Amazonian birds from these Várzea forests, we see similar patterns, always distinct lineages in the west, unique lineages in the different drainage basins in the east, we see evidence of cycles of isolation and contact in central Amazonia, as if this was a region that from time to time became inhospitable for these birds, like without flooded environments. And also we see that eastern populations have a stronger signal of recent variation in demography, showing that these habitats changed more through time. So finally, trying to put everything together, we think we can try to understand landscape evolution and the assembly of biological communities in different parts of Amazonia. This is a paper published last year as well. It shows a numerical experiment that incorporates surface and geodynamic processes inferred from the sedimentary record. And the interesting thing about this model is that it also tries to translate the geomorphological context to biomes, to different habitats in Amazonia. So this is the green is terra firme, várzea forest, igapó forest, and grasslands. And what the model shows is that landscapes of Western Amazonia sedimentary basins are very dynamic until recently, mostly the Solimões sedimentary basin here. There are long-lived upland forests in the shields here in the base of the shields, like the Guiana shield and the Brazilian shield, and in the base of the Andes. And
Camila,
yes.
sorry, you have 2 minutes, okay?
Okay, thanks. And then the history of the várzea in the west and igapó in the east are quite different. So this is a general animation of their model, showing what the evolution of the várzea in Western Amazonia. And the igapó has not appeared yet. Now igapó starts appearing at some point. And you can see that the West is quite dynamic in this region. When we translate this to habitat stability in the last 4 million years, we can see that the most stable regions are the yellow ones. All this purple area has been greatly unstable in the recent times. So a large portion of Amazonian landscape is recent and is undergoing recent change. Precipitation and geomorphology interact through time, determining expansion or retraction of the distinct Amazonian habitats. And a very interesting correlation that we found in this paper that we need to explore more is that we see a pattern of relationship between the phylogenetic diversity of bird communities and this pattern of stability or instability. So that communities with species that are less related than expected are found in stable areas, and communities with species more related than expected are found in unstable areas, showing a possible relationship between landscape evolution and the assemblage of biological communities. So the vast Amazonian diversity is a result of the interaction between climate, causing isolation and local extinctions, and rivers acting as temporally dynamic barriers that will constrain dispersal in different ways through time, and also in different ways for taxa with different ecological characteristics and different habitat affinities. So these evolutionary processes that originated and maintain Amazonian diversity can help us understand better the possible impacts of anthropogenic change, as Amazonia is probably going to change a lot in the next few years. So I want to thank the biological collections. The collaboration of biological collections in this project is very important because we know very little about Amazonia, and being able to use samples from several different collections in joint projects is essential for doing this work. And also all the funding agencies that have been helping us on our research. Thank you.
Okay, thank you very much, Camila. It's really wonderful, your presentation and the work that you have been doing. And thank you very much also for being here and accept our invitation. So I want to tell to everybody that you can make questions in the Q&A chat. So the first question is, the biogeographic patterns found in your study have also been found in other biological groups?
Yeah, it's a very important question. The studies in birds for some time have been criticized because they were the first ones to come out, and everybody was saying, oh, just because these are the areas of endemism for birds, will they be the areas of endemism for everything? Probably not. But the problem is there are a few groups for which we have good taxonomy so that we can find out which are the areas. For frogs, there is a paper published showing some congruence, but not completely congruence to the bird pattern. And for other groups, we are doing standardized surveys to try to understand how species change through time. But it's amazing how many new species appear in each field expedition. So it's very difficult to characterize.
Okay, thank you. This next question is there an explanation to why the oldest lineage persists in climate stable areas, but more recent lineage did not?
It's probably a matter of you having extinction at some point in these regions that are very unstable. And then you erase everything, the young and the old lineages. And then from there on, you have the presence of only the lineages that that originated after that event. So that you don't have the old lineages anymore. And then you start having only the recent ones that originated after the perturbation happened. So regions like the shields and the base of the Andes that seem to have been stable for a long time have greater probability of having places in which old lineages could have remained for a long time, while very unstable areas like lowland, Western Amazonia would have lost all the lineages at some point, and then being the diversification process is starting again there.
Okay, Salva, how we are with time for more questions?
I think we can make 2 question more because we have the coffee break, so there's no problem now. And I think there is a lot of questions, so maybe we can, yes. All the people enjoy the talk, so we can have a few minutes to talk about something like that.
Mm-hmm.
Okay, yes. Okay, there is a question that I think that is not related with your presentation, how the oil industry affect the wet habitat in the Amazonian. I think that may be like...
But it's very interesting. Not only the oil industry. I mean, the use of water is not a big problem. Amazonia has lots of water, but the alteration of river flow is very important. So anytime that you use the river water, you make a dam, for example, to collect water for something, for an energy generation or for anything else, you disrupt this annual cycle of flooding of the rivers. And this is very harmful for all the biota associated to the flooded environment and the terra firme forest biota that sometimes depends on this biota from the flooded environments for eating. So it's everything that you deal with in Amazonia has to be seen with caution because the system is largely interlinked.
Okay, there are a lot of questions and are very interesting. What about the extinct species? There are any way to prove if they were experiencing similar patterns?
The only thing we can do is reconstruct the distribution of ancestrals on our phylogenetic trees. Unfortunately, the paleontological record of birds in Amazonia is very poor, so we cannot know very well. But from the general optimization of distribution of ancestors, that's the general pattern we can take of it is that most phylogeny is optimized for the upland birds. Most phylogeny is optimized ancestors in the base of the Andes, in the Guiana Shield, and in the Brazilian Shield. So this should have a bearing in real life, but we don't know details about these distributions of the ancestors.
Okay. And the last question is, hi, about the biogeographical approach, how did you codify the different environments and implementing them in the phylogenies?
This is we can give a whole presentation about how to do that. But what I started noticing that was very important was that you should not do. We should not try to put flooded forest birds and upland forest birds in the same optimization, because they respond to completely different environmental histories. So when we study upland forest birds, as most lineages are delimited by the large rivers, it's very straightforward to use the areas of endemism as units, as biogeographical units. But when we study flooded forest birds, so far we haven't optimized, because flooded forest birds we have doing studies so far, our phylogeographical study is very recent, so we are on the population genomics realm, not doing optimization yet. But we have noticed that the first separations in these populations correspond to Eastern and Western Amazonia, and Western Amazonia seems to have had long-lasting and stable populations, where Eastern Amazonia has populations that have gone through demographic change recently. So we end up having to mix together several kinds of approaches in order to reconstruct these histories in different time frames. And I think that defining the areas for biogeographical optimization is a very important step of any biogeographic study, and it does not have to be the same in all of them. It depends on your question, on the distribution of your lineages, and specifically in what you want to do. With my students many times, we finish our work and we look again and say, maybe we should change the areas, because this is not addressing the question the way we wanted to. So it's a complicated issue that should be seen with a lot of caution. It's not possible to like take from your drawer, oh, these are the areas, so let me apply them to my group. Each group will have specific ecological characteristics and specific distributions, and will be interesting for asking specific questions, and the areas need to be defined accordingly.
Okay, thank you very much, Camila, for your time. Also, keep hearing the meeting if you can, to see the other presentations. If not, you can see them later in the YouTube channel. And we have 10 minutes of break, and we are going to start for the presentations at 11:40.
Can I answer questions here typing?
Yes.
Right.
During the break?
Yes,
okay.
Yeah. Okay.
Thank you.
Thank you very much.
Thank you very much again for the invitation.
To all the people that asked the questions. It's a very nice talk, and I think it's produced that kind of interchange. So we're happy.
Thank you. Okay, so I'll turn my camera off and we'll just type some answers.
Okay, thank you.
Thank you. creo que ya podemos dar inicio a la siguiente parte, ¿verdad, Nico? Okay, thank you for staying with us. And I think after this short break, that you can take some drinks and eat something. We are now ready to continue. So, Nico?
Okay, can you hear me, Salva?
Perfectly.
Okay, thank you. Okay, our next speaker is Ángela Mendoza-Henao, and she's from Departamento de Zoología, Instituto de Biología, Universidad Nacional Autónoma de México. Her presentation title is Hypotheses and Drivers for Diversification of Andean Tetrapods. She will be presenting in Spanish, and she will be answering questions in English and Spanish. So you can write questions in English and in Spanish. Thank you very much. And Angie, you are welcome.
Thank you. Gracias a todos. Voy a compartir pantalla. ¿Me dicen si se ve?
Perfecto.
Ok, bueno, pues muy buenos días. Muchas gracias a todos los asistentes. Muchas gracias a Nicolás y a todos los organizadores de este evento por invitarme pues, a dar esta presentación. Espero ser breve. Vamos a ver si lo logro, porque pues este es un tema muy grande, que son justamente cuáles son los procesos que han tenido que ver pues, con la diversificación de tetrápodos andinos. Voy a enfocarme principalmente en tetrápodos, aunque obviamente hay muchos estudios que se han hecho en invertebrados, en plantas, pero pues digamos que les un poquito más a esta parte, entonces mi charla va a estar enfocada un poco en esto. Entonces, pues para empezar, digamos, la base de la biogeografía y también de la macroecología es que la distribución de la diversidad de las especies no es igual en todo el planeta. Tenemos unas regiones que presentan una mayor diversidad que otras. Eso digamos que es como el común denominador de los trabajos de sobre todo supongo que de esta sesión. Y la región neotropical pues obviamente tiene mucha más diversidad que las regiones templadas. Pero dentro de esa región tropical tenemos algunas regiones que presentan una mayor diversidad que otras, y una que resalta mucho la atención, inclusive dentro del mismo trópico, es la región norte de los Andes, que es la zona aquí que está en rojo más oscuro. Esa zona pues ha sido, digamos, de gran interés porque presenta una extremadamente alta diversidad de muchos organismos. Entonces, ha sido, digamos, de interés para saber cuáles son los procesos que están dando lugar a esta alta diversidad de especies. Entonces, ha habido, digamos, varios trabajos que han estado tratando de sintetizar algunos de los procesos, y digamos aquí quiero traer a cotación un trabajo realizado hace 4 años en el que resumen, digamos, en términos de procesos de especiaciones, procesos de extensiones, procesos de colonización, qué es lo que está ocurriendo tanto en las zonas de Andes en comparación con zonas de Andes. Entonces hay 4, digamos, términos o hipótesis que se han establecido. Una es que los Andes son un nido de diversidad, en el que las especies de los Andes presentan unas tasas de diversificación un poco más altas que las zonas que no son andinas, pues entonces eso es lo que está provocando una alta diversidad. La otra es que los Andes es un museo de diversidad, un museo de especies, es decir, que mientras en otras regiones no andinas los procesos, digamos, ocurren extensiones continuamente, en los Andes no. Entonces esto hace que al final el valor absoluto de especies siga siendo mayor en la zona andina que la no andina. Esto respecto a los procesos de especiación y extensión, pero también podemos ver respecto a colonización, porque no solamente existen procesos de diversificación dentro de los Andes, sino respecto a regiones aledañas a los Andes. Entonces uno es el tiempo de especiación, que dice que dentro de los Andes han ocurrido los procesos desde hace más tiempo que en las regiones contiguas. Entonces, la región contigua en este caso empezó a diversificar solamente recientemente en comparación con la zona andina. Y la última, que es la del reactor de especies, que establece que los Andes tiene un algo que hace que las especies de otras regiones sean atraídas a ese sector. Eso respecto a digamos, a los procesos intrínsecos, digamos, de las especies per se. Entonces, digamos, un trabajo muy reciente, que es muy interesante, que justamente trabaja con un par de estas cosas, que es justamente lo de si son especies, si son cunas o si son museos de especies, identificó entre las montañas cuáles son las que tenían principalmente especies cuna, eran cunas de especies, que eran las zonas verdes, zonas museo, que son las zonas moradas, y las que tenían ambas. Y como pueden ver, el norte de los Andes, en esta región de aquí, comparada con todas las demás montañas del mundo, presenta, digamos, los dos efectos de una manera muy alta. ¿Qué quiere decir? Que en esta región están ocurriendo muchísimos procesos de diferente nivel que están dando lugar a esa diversificación. Entonces, con un compañero, que es Juan Carlos García, estamos trabajando en una especie de resumen pequeñito para identificar cuáles son estas, digamos, las causas que están dando lugar a que aquí en esa región haya mayores especiaciones, menores extensiones y posiblemente mayores procesos de colonización. Entonces, pues, lo estamos separando principalmente, digamos, en 4 grandes elementos de la estructura de los Andes. Una es la forma, otra es digamos, la forma en términos generales, otra es la irregularidad topográfica, el clima y los cambios geológicos. Voy a pasar un poquito en cada uno de estos. Entonces, el objetivo de este trabajo es pasar un poquito en cada uno de estos 4 elementos. Les voy a presentar algunos ejemplos para que veamos cómo estos pueden estar afectando lo que les comenté de los procesos de especiación y extinción. Entonces, el primero, que es la forma, es, digamos, el proceso estándar, es el proceso como en el que no hay ningún proceso detrás, simplemente es como distribución aleatoria de las especies, que establece que, dada una distribución regular, aleatoria de las especies a lo largo de un dominio, en este caso sería una montaña, cuando vemos los valores de diversidad a lo largo de esa elevación, nos va a dar un patrón en el que la mayor riqueza va a estar ubicada en el punto medio. Eso es simplemente un proceso aleatorio, y este es, digamos, el patrón, digamos, el modelo nulo para la mayoría de los estudios en diversidad en las zonas andinas o en las zonas montañosas en general. Entonces, si un grupo, la diversidad de un grupo coincide con este proceso, indicaría que no existen otros procesos más allá, que simplemente es un efecto geométrico. Entonces, cuando vemos algunos ejemplos, estos ejemplos en anfibios y este ejemplo en aves, vemos que digamos, la línea punteada es el intervalo en el que sería estrictamente un proceso de efecto de dominio medio, y vemos que tanto en ranas de cristal como en las ranas de desarrollo directo de Pristimantis tienen la diversidad es ligeramente mayor que la esperada por ese efecto geométrico. ¿Qué quiere decir? Que sí hay un proceso, que sí tenemos como ese impacto de la geometría, pero entonces también tenemos otros procesos más allá. Cuando lo vemos en aves, vemos que este patrón se observa en las especies que son endémicas de la región andina, pero no en las especies que están en los Andes, pero presentan una distribución más amplia. Entonces, son procesos relacionados directamente con especies únicamente presentes dentro de la región andina. Y respecto a entonces, ya cuando nos ponemos a meterle un poquito más de complejidad, vemos, por ejemplo, el efecto de la irregularidad topográfica, en el que a lo largo de la cordillera, pues no es que las especies se puedan encontrar en todas las regiones, sino que hay unos límites. Pueden ser que sean unos valles, pueden ser zonas más secas, zonas más áridas, que entonces pueden estar limitando la dispersión entre algunas especies, y está generando, pues, el aislamiento geográfico y posteriormente, pues, procesos de especiación dentro de estas regiones. Digamos, estas irregularidades en la zona andina se han evaluado, digamos, de diferentes formas. Este es un trabajo de uno de los colaboradores de este simposio, en el que se identifican, a partir de la distribución de algunas especies, algunas barreras y algunas regiones que presentan diferencias entre las estructuras de las comunidades, entre las estructuras de las especies, en las que podemos entonces identificar cuáles son unas regiones biogeográficas dentro de la zona de los Andes. Y aquí, digamos, un ejemplo un poquito más pequeño, no solamente dentro de esas grandes bioregiones andinas, sino que dentro de esas mismas podemos encontrar algunos procesos de aislamiento, de procesos de separación. Aquí tenemos el ejemplo de tres ranas, de tres ranas que se encuentran en la misma cordillera, sería en la misma bioregión, que están ubicadas en diferentes regiones. Y vemos que en los puntos de diferente color, esto lo hicimos con genes nucleares hace un tiempo, vemos unos valores de sustitución bastante altos. ¿Qué quiere decir? Que entre esta región de acá y esta región de acá, que son no más de 60 kilómetros, entonces ya estamos viendo el proceso de aislamiento entre poblaciones y posiblemente estamos viendo posibles procesos futuros de especiación. Estamos viendo entonces posiblemente microendemismos dentro de cada cordillera y esto nos da una idea de lo que serían procesos de cunas de especies, que es lo que les estaba comentando al principio. Otro ejemplo sería, digamos, este trabajo que hicimos con aves, que trabajamos con DNA barcoding. El objetivo era, digamos, otro, que es simplemente identificar secuencias de DNA que sirvieran como barcoding, pero entonces trabajamos principalmente con especies de los Andes, 147 especies, principalmente colibríes, y encontramos que encontramos algunas especies en las que la morfología no coincidía con la identificación morfológica no coincide enteramente con la identificación aquí con el DNA, y ese porcentaje de divergencia fue mayor para las especies andinas que lo que se había reportado para especies de aves en la zona de Norteamérica, como en las zonas de Brasil y en las zonas de Australia. Esto fue principalmente para especies colombianas, y les quiero traer algunos de esos ejemplos que presentaban esas altas divergencias. Encontramos, por ejemplo, este colibrí, que entre estas dos localidades, que son las figuras diferentes, tenemos porcentajes de hasta 6% de divergencia entre las dos. Y en este búho también encontramos unos valores de divergencia extremadamente altos entre dos cordilleras diferentes. Entonces aquí podemos estar viendo procesos de diversidad críptica, de especies, posiblemente pueden ser procesos de especies hermanas que están divergiendo entre diferentes sistemas montañosos. Y dentro de los mismos sistemas montañosos también encontramos procesos de unos aislamientos extremadamente altos. Entonces, el caso de este colibrí, Xistes geoffroying, encontramos en localidades extremadamente cercanas, vemos que un valor de divergencia en citocromo oxidasa extremadamente alto, 12.6% de divergencia. Eso es absurdo. Entonces, aquí que estamos viendo, que dentro de una sola región tan pequeña también estamos observando procesos de aislamiento bastante profundos. Y aquí quiero completar con este último ejemplo, en el que tenemos el valor de divergencia no está tanto relacionado con valor entre cordilleras, sino que la secuencia divergente es una secuencia que está ubicada en términos de elevación un poco más altos que los otros individuos de esa misma especie. Entonces, aquí quiero saltar entonces al segundo proceso, entonces un proceso ya más respecto a la elevación, que es el clima. Entonces, ¿qué es lo que está pasando en la región andina? Que no está ocurriendo, o que está ocurriendo tal vez menos en otros sistemas montañosos. Cuando comparamos las especies de zonas templadas con las de zona tropical, una hipótesis que se está evaluando mucho, es que las especies de las zonas templadas tienen un rango elevacional mucho más amplio que las especies tropicales, lo que permite que estas puedan tener una diferenciación más estrecha, una mayor diferenciación. Asimismo, la ubicación ecuatorial de las montañas nos permite que tener hábitats, no tan agrestes como las nieves perpetuas, hasta elevaciones un poquito más altas, y eso sumado con las elevaciones altas de la cordillera de los Andes, puede dar lugar a que haya, un proceso de mayor divergencia. Entonces, ¿esto a qué se debe? Principalmente, pues lo que les digo, la posición en el Ecuador permite que las condiciones del clima a lo largo del año sean estables, sean muchísimo más estables en las regiones tropicales que en las regiones templadas. Y digamos, es un proceso eso es un trabajo que ha venido agarrando más fuerza digamos, en los últimos años. Uno cada vez ve más estos estudios en los que diferencian, en los que se evalúan las temperaturas máximas y mínimas de las especies para ver si efectivamente esas especies tropicales de montañas tropicales tienen este patrón. Y efectivamente los trabajos están dando lugar a eso. Y aquí lo que yo quiero, digamos, hacer énfasis es, bueno, este proceso más allá, digamos, de los límites de temperatura, cómo está afectando en algunos, algunos caracteres, como por ejemplo los rasgos de cortejo en algunas especies, en este caso sería ranas de cristal. Estoy bien de tiempo, sí. Entonces lo que hicimos, lo que está, es un trabajo que acabamos de someter, en el que vimos si la variación no solamente en temperatura, sino en vegetación, va a estar afectando los rasgos que se están afectando el variamiento entre las especies. Y esta variación comportamental puede estar promoviendo también los procesos de especiación dentro de los sistemas montañosos. Porque entonces, claro, las especies ya digamos, tienen problemas de reconocimiento intraespecífico, entonces pues ya ahí, digamos, se aceleran los procesos de especiación. Y eso, pues, cuando lo vemos en ranas de cristal, cuando vemos el patrón de diversidad, no solamente que es muchísimo mayor en las zonas montañosas de lo esperado por azar, sino que vemos que en unas de las subfamilias, que es Centrolenine, presenta una mayor diversidad en la zona andina que la subfamilia que es más presente en las zonas bajas, que es Yalinobatrachia. Y asimismo vemos que el endemismo, el que es identificado aquí por el área por sitio, es bastante alto en las zonas andinas, y asimismo el endemismo filogenético es...
Angie, discúlpame, te quedan 2 minutos, ¿listo?
Muy bien,
2 minutos.
listo, muy bien. Entonces si identificamos esto, cuando lo vemos respecto a los cantos de las especies, entonces aquí no voy a ir muy adelante, podemos detectar que tanto la vegetación como la temperatura sí están afectando en algunos rasgos, que en este caso serían la frecuencia pico, la tasa de notas y la tasa de pulsos, mientras que la temperatura está afectando en la frecuencia pico y la amplitud de la frecuencia. Y esto es más visible en los grupos, como les dije, en la familia que está más presente en la región andina. Entonces, ¿qué estamos encontrando aquí? Que en estas regiones puede haber tanto, en la región andina podemos ver el proceso de nidos de especies y tanto como museos. Y el último, pues, son esas figuras que yo creo que vamos a ver mucho en aquí veo a Carina Hoorn riéndose, estas figuras que vamos a ver mucho en este simposio, que es los cambios geológicos, que no solamente, digamos, estos procesos no son constantes, sino que hay variación en la región andina que está dando procesos tanto de aislamiento. Entonces voy a hacer más énfasis en el impacto de la subida de los Andes, pero también tenemos procesos pleistocénicos. De los ciclos pleistocénicos que generan, digamos, variación en los climas en las zonas altas y bajas, que está dando lugar a procesos de mayor diversidad y procesos de altas especiaciones dentro de estas regiones. Entonces, como no tengo mucho tiempo, voy a saltarme este ejemplo y voy a terminar con este último, que fue un trabajo que hicimos con un grupo altamente diverso, en el que vimos cuál es el área de origen y cuál es digamos, cuando comparamos cuál es el área de distribución de las especies, y aunque tuvimos un árbol que todavía está incompleto, encontramos que, digamos, dos cosas importantes. La primera es que el norte de los Andes podría estar considerado el origen de muchas de estas especies, y segundo, que las especies que están presentes en esta región presentan, digamos, se agrupan de cierta forma. Entonces, ¿qué quiere decir? Que tenemos especies que el grupo se distribuye en los Andes, y asimismo dentro de esa misma región hay altos procesos de especiación. Bueno, esto también me lo voy a saltar porque ya se me acabó el tiempo. Entonces, pues como en resumidas cuentas, tenemos dos sets de procesos, unos procesos que son intrínsecos de los elementos de especiación y de extinción y de colonización, y otros, digamos, los elementos que están afectando a esos procesos. Entonces, la idea es como hacer unión de estos dos conjuntos de elementos para tener en cuenta qué es lo que está haciendo que esta región de los Andes sea tan alta, tan altamente diversa. Y bueno, pues me hubiera gustado poder hablar un poquito más, pero el tiempo es oro, y muchas gracias a los que vieron esta charla, y cualquier pregunta espero tener tiempo para responderla.
Listo, Angie, muchísimas gracias por la charla, muy interesante, y sí, obviamente con todas las charlas nos encantaría tener al menos 5 minutos más.
Sí.
Listo, entonces aquí tenemos 2 preguntas. To Dr. Mendoza from Antonio Aguiar, is it possible to suggest that a timescale pattern of divergence of distinct endemic areas of Andes?
Pues si nadie me entiende el español, la verdad podría decir que uno para hacer una escala de tiempo uno tiene que hacer unas calibraciones de los árboles, y esto es posible. De hecho, en el último ejemplo lo que hicimos también fue calibrar para identificar cuáles son los elementos, cuáles son los tiempos en los que pudieron ocurrir diferentes procesos. Y a ver si entiendo la segunda mitad de la pregunta. Y sí, claro, pues digamos uno lo que hace cuando hace reconstrucción de áreas ancestrales, por ejemplo, es hacer esas calibraciones, compararlas con algunas de las regiones que serían de interés para el estudio. Entonces, en este caso, lo que yo quería ver era si el norte de los Andes tenía alguna diferencia en el origen respecto a otros grupos, pero entonces, si uno puede hacer, por ejemplo, con las otras áreas geográficas dentro de los Andes, pues perfectamente se puede hacer.
¿Qué tanto piensas que el interplay y entre la estructura montañosa de los Andes, la Amazonía, y puede explicar la diversidad tan grande del norte de los Andes?
Esa es otra cosa muy interesante, porque ese sería el proceso de colonización hacia los Andes, que se ha evaluado en otros grupos. Creo que en ranas dardo, bueno, serían sí, en dendrobatios, porque no todos son ranas dardo, se ha encontrado ese efecto de que no solo las especies dentro de los Andes tienen procesos de especiación dentro de la misma, sino que especies amazónicas llegan a los Andes y ahí divergen y ahí especian. Eso digamos, es un ejemplo, pero creo que hay muchos más.
Ok. Salva, ¿tenemos tiempo para alguna otra pregunta más?
Sí, yo creo que una pregunta más. Y Ángela, estás en Discord, ¿verdad?
Sí.
Si alguien quiere contactarse con Ángela, ella está en Discord, entonces ahí pueden seguro preguntarle más cosas en algún momento.
Bueno, ¿hay algún patrón reconocido de diferencias en estructura genética entre miembros de Terrana a lo largo de los Andes, o presentan en general historias evolutivas similares?
Sí, hay diferentes procesos, hay diferentes historias que hemos encontrado. Igual, como te digo, es necesario, digamos, complementar un poquito con un mayor número de especies para que esos procesos sean estables, pero sí hemos encontrado principalmente, digamos, que las especies andinas radian dentro de los Andes, pero también hemos encontrado algunos casos en los que hay migraciones de las especies, tanto, digamos, del norte de los Andes hacia la zona de Costa Montana en Venezuela o hacia los Andes centrales, o entre las especies que están en el norte de los Andes y el Chocó, o el norte de los Andes y Amazonía. Pero y obviamente pues algunas que han ido hasta Centroamérica, que ha habido múltiples procesos de colonización hacia Centroamérica. Entonces, sí, digamos, tenemos una heterogeneidad de historias que han ocurrido en este grupo.
Ok, listo, Angie. Muchísimas gracias por tu tiempo, por aceptar nuestra invitación, y quédate con nosotros siguiendo las demás presentaciones. Muchas gracias. Felicitaciones
Muchas
por tu
gracias a
trabajo.
Gracias.
Sí. Okay. Esperen que está un poco demorado. Ok. Salva, ¿se puede ver la presentación?
Perfecto.
Ok.
Sí.
And our next speaker is Jonathan Pelegrin Ramírez. He is from Área de Biología y Ciencias Ambientales, Universidad Santiago de Cali, y Departamento de Biología de la Universidad del Valle, Colombia. His presentation title is Evolutionary, an Ecological Story of Falcons, Macroevolutionary Analysis with an emphasis in the Neotropics. I just want to make sure, Jonathan, you will be presenting in Spanish, right?
Right.
Or, okay, so Jonathan will be presenting in Spanish and answering questions in English and Spanish. Okay, Jonathan, you are welcome. Let's start.
Ok, muchísimas gracias. Voy a compartir por acá. Ahí todos ven la presentación, ¿cierto? Sí.
Perfecto.
Perfecto. Sí. Ok. Bueno, primero que todo, muchísimas gracias a la organización y al, digamos así, el comité científico del evento, pues por esta invitación y felicitarlos también, pues, por esta iniciativa tan importante, digamos así, en la divulgación de la biogeografía y la evolución, y que pues la pandemia no sirva de barrera para eso. Bueno, el día de hoy quiero presentarles, digamos, una pequeña parte, digamos, de lo que he investigado durante el desarrollo de mi doctorado, que básicamente pues se enfocó en las aves no paseriformes, y aquí específicamente quiero hacer un énfasis en los halcones. Ya vamos a entender por qué los halcones, ¿sí? Y básicamente lo que intentamos hacer es reconstruir la historia evolutiva y ecológica de los halcones con base en un análisis macroecológico y macroevolutivo que vamos a ver ahora. Entonces, para que, digamos, nos contextualicemos primero, ¿cierto? A la manera de introducción. Entonces, digamos que hay una pregunta muy importante que nos podríamos hacer y es, digamos, desde un punto de vista filogenético, desde una perspectiva filogenética, pues, de alguna manera, cómo las especies logran colonizar los biomas emergentes, porque ya veíamos en charlas anteriores que, ¿cierto?, que hay unas dinámicas terrestres, ¿cierto?, relacionadas con procesos climáticos, procesos geológicos, diversos tipos de procesos, como los que veíamos también ahora en los ríos, que de alguna manera, pues, hacen que los biomas tengan una dinámica histórica importante. Entonces, obviamente, las especies también responden a esa dinámica, y entonces es importante plantearnos la pregunta de, bueno, ¿cómo es que ellas logran colonizar esos biomas que emergen o se transforman? ¿Sí? Entonces, en ese orden de ideas es fundamental definir el concepto de conservacionismo de nicho, o niche conservatism, que básicamente se define como la tendencia que tienen las especies a retener sus características o caracteres ecológicos ancestrales. Entonces, digamos que para verlo de una manera más didáctica y de una forma más gráfica, supongamos que aquí tenemos un patrón de parentesco de especie, en este caso, pues colibríes, obviamente es hipotético, ¿sí? Supongamos que este es un patrón filogenético. Entonces, digamos que ellos tienden a evolucionar o a diversificarse, reteniendo esa característica, o en ese caso, características que les permiten asociarse a un entorno concreto, ¿sí? Entonces, aquí vemos, por ejemplo, por poner un ejemplo, ¿cierto?, 4 especies que descienden de una que está asociada a ese entorno, entonces ellas heredan, por lo tanto, pues la característica de su ancestro, ¿cierto? Pero entonces, ahí donde viene el tema de cómo es que logra en un momento puntual, una especie logra, ¿cierto?, acceder o adaptarse, digámoslo así, al uso de un recurso o un ambiente distinto, ¿sí? Entonces ahí es donde ya hablamos de colonización, y entonces obviamente, digamos que esos eventos se asume, ¿cierto?, pues por lo que se ha visto en diversos estudios con diversos grupos, se asume que es mucho menos frecuente y que lo más frecuente es lo primero, ¿sí? Entonces, digamos que en otro contexto, ¿cierto? Algunos autores lo que han postulado como críticos y colaboradores es que el concepto de conservacionismo de nicho puede ser, digámoslo así, extendido al concepto macroecológico de bioma, ¿sí? Ya sabemos que los biomas pues son estas unidades macroecológicas muy grandes, definidas por variables bioclimáticas, y entonces, claro, digamos que esos recursos ecológicos están asociados a un bioma, y por lo tanto, también podremos decir con cierta validez que esos nichos, o esa tendencia al conservacionismo de nicho, puede estar ligada a lo que llaman algunos autores, como precisamente Cris y colaboradores, el conservacionismo de bioma, o bioconservatism, que básicamente, digamos que es una ampliación del concepto de conservacionismo de nicho, pero en este caso al concepto del bioma, y entonces, pues en este caso, las especies retendrían la ocupación biómica de sus ancestros, digámoslo así. Entonces, en ese orden de ideas, ¿cierto? Vamos a preguntarnos precisamente con base en ese contexto teórico, pues cómo fue el proceso de colonización, digámoslo así, de los biomas emergentes en los continentes, ¿sí? Entonces, digamos que una de las grandes dificultades que tuvimos para poder evaluar esto es que, como vimos en la diapositiva anterior, es fundamental tener digamos, una filogenia, una resolución filogenética importante, pues ojalá lo más completa posible, ¿cierto? Entonces, de ahí viene el surgimiento de por qué lo hicimos con halcones, ¿sí? Entonces, básicamente porque los halcones, digamos que tenemos una ecología muy bien conocida de ellos, todos sabemos que son especies que de alguna forma forman piezas claves en los ecosistemas y en los entramados ecológicos, ¿cierto? Digamos que el orden falconiformes actualmente está relativamente bien definido, ¿cierto? A partir de estudios moleculares, se tiene una filogenia completa a nivel de especie, publicada por Fuchs y colaboradores en el 2015, y además de eso, desde el punto de vista biómico y ecológico, los halcones, pues, o el orden falconiformes y la familia Falconidae, es una familia con distribución global y ecológicamente los encontramos en todos los biomas del planeta. Desde las selvas tropicales, ¿cierto? Por ejemplo, aquí en Sudamérica, como lo vemos allí con el Petotenes calcinans, que es el halcón culebrero, hasta el Falco rusticolis, por ejemplo, que lo vemos en zonas nivales. Entonces, digamos que este grupo lo utilizamos para precisamente tratar de responder a esa pregunta que nos hacíamos de cómo es que las especies logran colonizar esos biomas. Entonces tenemos, digamos así, caracterizados 10 biomas, de acuerdo a las propuestas, digamos, de Whittaker, Walter y Hernández Fernández en el 2001. Entonces, para evaluar esto inicialmente lo que hicimos fue codificar digámoslo así, lo que llamó Hernández Fernández y Verba en el 2005, el BCI. El BCI significa Biomic Specialization Index, que es la medida de qué tan especializada está una especie en uno o muchos biomas. Entonces, bueno, con base en la distribución geográfica y unos criterios metodológicos que pues no va a entrar en detalle porque no tenemos tiempo suficiente, pues codificamos esa presencia de cada una de las especies, de cada una de las 66 especies en cada uno de los biomas, ¿sí? También, entonces, teniendo esta codificación, realizamos unos análisis utilizando Biogeography, unos análisis biogeográficos, utilizando los biomas como áreas geográficas, y también utilizando las filogenias calibradas para los halcones. Y en ese caso, es importante destacar que el análisis nos permite incluir información temporal, digámoslo así, y espacial de los biomas también, porque hay que tener en cuenta que cada uno de los biomas, ¿cierto? Cada uno de los 10 biomas que tenemos en el planeta, pues tiene una edad distinta porque obedece a unos patrones paleoclimáticos históricos diferentes. Entonces, la ventaja de este análisis que nos permitió introducir una matriz de, digámoslo así, de historia de los biomas en la Tierra, ¿sí? Que obviamente la inferimos a partir de bibliografía y a partir de estudios paleoclimáticos. Entonces, bueno, ¿qué obtuvimos? ¿Cierto? Aquí vemos un poco simplificado el árbol, porque obviamente es un árbol de 66 especies, entonces voy a hacer énfasis como en algunos aspectos más relevantes. Estos son como la base del árbol, ¿sí? Aquí vemos, digámoslo así, las dos subfamilias, que serían el Petoterinae y Falconinae, ¿sí? Y Falconinae a su vez se divide en dos tribus, que es Polyborini y Falconini. Entonces, digamos que las tendencias filogenéticas en la ocupación de los biomas, vemos allí que nos reconstruye con mayor probabilidad una ocupación generalista, asociada el ancestro a 3 biomas, que en este caso los que vemos con colorcito morado y los 2 verdes, ¿sí? Que básicamente son los biomas que vemos acá. El morado es la pluvisilva o bosque lluvioso tropical, como el que encontramos, por ejemplo, en el Amazonas, representado. el verde claro son los bosques secos tropicales y el verde oscuro son las sabanas, ¿sí? Como por ejemplo en las zonas de Caatinga aquí en Sudamérica, ¿sí? Entonces, vemos que el análisis nos reconstruye una ocupación biómica y biogeográfica asociada en el ancestro de los falconiformes a estos tres biomas. Entonces, ir a una condición generalista que, como podemos ver acá, se conserva, ¿sí? en la parte inicial de la diversificación del grupo, ¿sí? Luego ya vamos a ver unos patrones interesantes en donde se pierden en algunos linajes, distribuciones, y en donde se ganan en otros linajes, distribuciones, ¿sí? Entonces, esta es como la tendencia inicial. Aquí lo vemos ya el árbol completo. Digámoslo así, tenemos todos los nodos con la reconstrucción de probabilidad de la ocupación biómica en cada uno de ellos. Y aquí vemos más o menos desde el Oligoceno, que es cuando de acuerdo a la filogenia, se estima el origen de lo que llamamos crown falconiformes, o digamos, o crown falconide en este caso, que es el grupo, digamos, que incluye todos los halcones actuales, ¿sí? Entonces vemos acá ¿cierto? Lo que les decía, una condición generalista, ¿cierto? Asociada a estos tres biomas que les decía, y vemos acá también cómo esa condición generalista relativamente se conserva, ¿cierto? Se conserva en gran parte de la filogenia, por lo menos en la parte inicial. Esto es bien interesante tenerlo en cuenta. Ya vamos a ver por qué. Entonces hay una conservación importante de la condición generalista en la ocupación biómica, ecológica, digámoslo así, en el proceso inicial de diversificación de los falconiformes, ¿sí? Entonces, pero entonces ustedes preguntarán, bueno, ¿y qué pasa con el neotrópico? Entonces, ahora vamos a hacer un énfasis precisamente en lo que sucede en el neotrópico, porque precisamente también este grupo tuvo un centro de diversificación neotropical. Entonces, de acuerdo al análisis que hicimos de cada uno de los nodos, encontramos una alta conservación de ese conservacionismo de bioma, precisamente. Con respecto digamos así, a la parte biogeográfica, digamos que lo que obtuvimos está en línea con otros estudios que se han realizado. En ese caso también se tuvo en cuenta en ese estudio de 2016, se tuvo en cuenta el registro fósil de los falconiformes. Y algo bien interesante es que este estudio precisamente también que hace una aproximación biogeográfica, incluyendo en ese caso datos fósiles, ¿sí? Reconstruye para el Oligoceno una diversificación muy importante, ¿cierto? De los crown group, que llamamos falconide, para Sudamérica, ¿sí? Y entonces en ese caso, precisamente en Sudamérica, en este proceso de Eoceno-Oligoceno, allí está, lo interesante es que esos tres biomas fueron muy importantes, y lo hemos visto en las charlas anteriores, ¿cierto? Por ejemplo, con las dinámicas que nos mostraban de la evolución de la Amazonía, esos tres biomas o la relación de esos tres biomas ha sido fundamental en Sudamérica. Entonces, vemos cómo, digámoslo así, los estudios empiezan como a encajar entre sí, y eso es bastante interesante porque nos crea un marco biogeográfico para la evolución de este grupo. Entonces, digamos que esa diversificación inicial en Sudamérica fue bien importante. Vemos aquí una diversificación grande en el neotrópico, que también estaría de acuerdo con lo que obtuvimos nosotros, y entonces, haciendo énfasis como en esa diversificación no tropical, aquí vemos lo que obtuvimos para los dos taxones, digámoslo así, principalmente no tropicales, que es la subfamilia Herpetoterinae, ¿sí? Que en donde encontramos, por ejemplo, el género Herpetoteres, que es muy común, y la diversificación interesante que vemos en Micrastur. Entonces, por ejemplo, aquí en el género Micrastur, ¿sí? Vemos una diversificación bien interesante aquí en el Plioceno-Pleistoceno, y precisamente vemos mucho protagonismo de estos biomas tipo bosque húmedo tropical y bosque seco, y precisamente este tipo de patrones los podemos explicar con algunos de los procesos que los otros conferencistas comentaban ahora, ¿cierto? Los procesos de fragmentación debido a cambios climáticos que se dieron hacia el final del Neógeno, precisamente en esa disminución gradual de las temperaturas, y que culminan en el Pleistoceno precisamente con todas esas grandes fluctuaciones de las glaciaciones. Entonces, eso es bien interesante porque a pesar, digámoslo así, de esa gran diversificación en el neotrópico, pues lo que se observó fue un alto conservacionismo de bioma en las especies. Entonces aquí vemos, y por su parte, ¿cierto? Digamos, todo ese gran clado, luego que digámoslo así, se desprende que sería lo que llamamos falconine como tal, y luego sobre falconini, que son los que se diversifican en el viejo mundo. Y es interesante esa diversificación en el viejo mundo, no que eso pues, nos daría para otra charla, porque digamos que estuvo muy asociada a la emergencia de ambientes abiertos, tipo estepa, tipo pradera, tipo sabana, en el viejo mundo. Entonces, eso fue bastante interesante. En términos de tendencias, aquí vemos un poco como las dinámicas de colonización ya graficadas para los falconiformes. Entonces aquí vemos, por ejemplo, en lo que llamamos
Jonathan,
templado.
Jonathan, discúlpame, te quedan 2 minutos, ¿listo?
Ok, vale, Nicolás. Entonces, en lo que llamamos el bosque templado vemos una temprana colonización, ¿sí? Hay una temprana colonización hacia más o menos Oligoceno-Mioceno, que básicamente estuvo relacionada con los ancestros del linaje de los herpetoterinos. Luego vemos aquí también ¿cierto? Un incremento importante hacia finales del Mioceno de la colonización de los bosques tropicales húmedos, ¿cierto? El bioma típico, por ejemplo, que encontramos en Amazonía, por ejemplo, en el caso de los del género Micrastur, donde vimos esa importante diversificación en el neotrópico. Luego tenemos un importante crecimiento, a su vez, de la colonización en ambientes tipo bosque seco tropical y tipo sabana, que precisamente tienen que ver con esa gran diversificación de la que les mencionaba, que se observaba en el viejo mundo para los falconinos, de género falco y poligueras, que de hecho fue el género falco el que más especies tiene en la actualidad. Luego ya hacia más o menos el Plioceno, Pleistoceno empiezan a consolidarse, debido a que la temperatura baja empiezan a consolidarse ambientes abiertos y muy secos, tipo desierto y tipo bosques mediterráneos, y entonces allí también ya se empieza a ver la colonización de ciertos linajes, específicamente dentro del género falco, como el linaje de falco con colos, y posteriormente, ¿cierto?, desde ambientes estacionales, tipo sabana, tipo ambientes abiertos y relativamente secos, se da la colonización de los bosques deciduos, ¿sí?, en linajes como columbarios y amurensis, que a su vez también entrarían posteriormente en las estepas y los desiertos fríos y los bosques boreales, y precisamente podrían dar origen a especies que son altamente generalistas, como Falco peregrinus, que en últimas tendría como, digámoslo así, las preadaptaciones debido a condiciones generalistas del linaje para poder finalmente entrar en las tundras, ¿sí?, que se consolidarían durante la era de hielo en el Pleistoceno. Entonces, claro, pues digamos que eso nos permite, desde un punto de vista ecológico, macroecológico y biogeográfico, reconstruir un poco la historia de las especies. Cuando analizamos un poco como las direccionalidades de la colonización, vemos aquí en colores los diferentes biomas, vemos destacados los verdes, ¿cierto? El 2 y el 2-3, que representan las sabanas, el 2-3, y el bosque seco tropical, el 2. Entonces vemos que a lo largo de la evolución de los falcónidos, estos dos biomas, digámoslo así, que constituyeron, digámoslo así, nidos, cradles de especies, son como pools de especies que de una manera fueron fundamentales en esos procesos de diversificación, en este caso particular de los falconiformes. Y precisamente esos biomas, ¿cierto? El bioma 2 y 3, ¿cierto? Aquí vemos en la tabla un poco las transiciones entre un bioma y otro, entre el bioma destino y el bioma fuente, y entonces vemos un poco que el conservacionismo de bioma es muy alto, es significativamente alto en biomas que se consideran climáticos extremos, ¿sí? Es decir, que tienen variables climáticas extremas, y eso es bien interesante porque eso ya nos dice que fisiológicamente, para yo vivir en un bioma climáticamente extremo, tengo que hacer unos ajustes, y eso constriñe mucho más el conservacionismo, porque digamos que la fisiología es fundamental para yo poder adaptarme ecológicamente a unas condiciones. Entonces, ahí vemos en los circulitos los dos biomas más destacados en la evolución de los falcónidos, ¿sí?, el 2 y el 2-3. Y finalmente, ¿cierto? Digamos que esos patrones de ocupación biómica para los halcones.
Jonathan, discúlpame, pero tu tiempo se acabó. ¿Puedes continuar y terminar la presentación lo más
un segundito,
rápido
no
que
está la
puedas?
última. Está la
Listo,
última.
dale.
¿Listo? Entonces, estos resultados que nos dieron, digámoslo así, pues fueron, digámoslo así, consecuentes con lo que se obtuvo en otros estudios para carnívoros, también exactamente lo mismo. Entonces, de alguna manera es interesante porque vemos que desde el punto de vista ecológico y macroevolutivo, pues, digámoslo así, la condición depredadora pues independiente que sean aves o mamíferos, pues generan unos patrones similares. Entonces, es bien interesante esa coincidencia. Entonces, finalmente, pues para aquí les presento unas conclusiones para que, digamos, podamos sintetizar en algunos puntos lo principal que se obtuvo de estos análisis. Entonces, fundamentalmente, el papel de los bosques secos tropicales y las sabanas y de las variables climáticas en ese conservacionismo de nicho. Y bueno, y muchísimas gracias por su atención. No sé, pues, pero ahí también por el tiempo y de nuevo, si hay alguna pregunta y hay tiempo, pues con mucho gusto.
Listo. Listo. ¿Puedes ver las preguntas, Jonathan?
Espera un momento, creo que tengo que dejar de compartir, me parece, porque no las puedo ver.
Ok.
Listo. Ya.
Listo.
A ver, preguntas por acá. Listo.
Hola, Jonathan, muchas gracias por la charla. ¿Has investigado cómo se relaciona el conservatismo de bioma y la adaptación a nuevos biomas con la diversificación de los linajes? Y si estos patrones también se observan en otras aves y o resto de vertebrados.
Ok, bueno, particularmente, digamos, yo me he enfocado mucho en los temas relacionados con aves, entonces, digamos que la gran dificultad que ha habido para aplicarlo a todas las aves es que, digámoslo así, carecemos de una filogenia por lo menos muy completa, hay unos super tris, pero todavía, digamos, faltan muchas especies y lamentablemente no funciona muy bien los análisis cuando hay muchas incertidumbres, digamos, filogenéticas. Entonces, digamos que hasta el momento se ha trabajado con halcones y con otro grupo que hemos trabajado es con galliformes, que tienen que hay filogenia relativamente muy completa, más del 75% de las especies. Entonces hemos trabajado con esos dos, ¿sí? Entonces digamos que aparte de este grupo de aves, pues básicamente estos dos que les menciono. Y en otro grupo de vertebrados, pues realmente son muy pocos los estudios que han hecho con conservacionismo de bioma. Por allí creo que hay algo, pero muy, muy distante en peces, obviamente adaptado a las condiciones acuáticas, y principalmente se ha enfocado todo esto del conservacionismo de bioma a las plantas, a grupos de plantas, y en artrópodos sé que hay algo también con arañas.
Ok. Hay una pregunta muy interesante que dice, gracias Jonathan por la presentación. Si las características de los biomas cambian a lo largo del tiempo, ¿por qué utilizar las propuestas de biomas y no áreas biogeográficas resultantes de la distribución natural de las especies? ¿Eso no incluiría un sesgo en las interpretaciones para el pasado?
Claro, el tema allí es que decidimos utilizar los biomas porque de todas maneras, precisamente, digamos, es más sencillo desde el punto de vista de la aproximación, o por lo menos tenemos más datos de conocimiento de, por decir algo, en determinadas localidades, qué bioma había en qué momento. O sea, digamos que tenemos conocimiento con respecto al paleoclima, de qué biomas había en la Tierra en determinado tiempo. Lo que sí es difícil estimar es el área, ¿cierto? Digámoslo así que, o digamos con mucha incertidumbre, había una subestimación del área que tendríamos en el pasado. O sea, digamos que, y en ese caso, digamos que el efecto del área, digamos, no lo tuvimos mucho en cuenta porque lo que nos interesaba era más, por decir algo, cómo el organismo intenta o tiende a conservar la característica de estar presente en unas determinadas condiciones. Y entonces digamos que en ese orden de ideas, pues, digamos que si el área es grande y se fractura, ¿cierto? Se fragmenta, entonces se genera un proceso de aislamiento reproductivo y luego pues, geográfico y comienza la especiación, pues ver cómo esas especies, digámoslo así, siguen estando ligadas a las mismas condiciones ambientales, digamos, independiente del área que había originalmente. No sé si me he hecho entender allí. Entonces, digamos que para nosotros fue más importante, digámoslo así, no es que no es importante, sino que pues, para nosotros en ese caso lo que queríamos responder era bueno, ¿cómo es la tendencia en la conservación de bioma y obviamente las características que subyacen a eso a lo largo de la filogenia y cómo se relaciona con los patrones de diversificación, digámoslo así.
Listo. Jonathan, muchísimas gracias por haber aceptado nuestra invitación. En serio, magnífica charla. Y también pues puedes seguir con las demás charlas y puedes responder estas preguntas en el
Sí, por
chat.
aquí voy a intentar aquí lo escribo. ¿Listo? No hay problema.
Listo. Vale, Jonathan, muchas gracias.
Ok.
Vale,
Gracias a ustedes, chicos.
Okay, our next speaker is Ubirajara Oliveira. His presentation title is Searching for Biogeographic Patterns in Data with Sample Gaps and Bias, something that is very interesting. He is from Centro de Censuramiento Remoto, UFMG, Associate Professor at the postgrade in the Modelaje de Sistemas Ambientales, IGC, UFMG, and Associate Professor at the postgrade in Zoología in ICB, UFMG. Ubirajara, you are welcome to start your presentation. Thank you very much.
Thank you.
Sorry, I forgot to say that he will be presenting in Portuguese and he will be answering questions in Portuguese and English.
Or the questions can be asked in English or Spanish, not problem.
Okay, perfect. Thank you.
Vou falar então um pouco sobre a busca de padrões biogeográficos em meio a dados com lacunas. A minha apresentação tá para... My presentation is working?
Yes.
Yes, it's working.
Yes? Okay. Então, vou falar um pouco sobre essa busca de padrões biogeográficos em meio a dados com uma série de problemas, como lacunas e viés de coleta, que é muito comum, principalmente aqui na região tropical. Então, nós vivemos na era da informação, na era dos dados. Existe uma disponibilidade muito grande de dados e uma disponibilidade muito grande também de meios para se analisar esses dados. Nós temos hoje em dia uma série de computadores pessoais que são capazes de processar grandes quantidades, grandes volumes de dados. Temos também softwares adequados para o processamento desses dados. Então, nessa era da informação, quando a gente pensa em dados de biodiversidade, nós temos, por exemplo, o GBIF, que é uma grande base de dados sobre informação de biodiversidade. Essa grande base de dados, atualmente, conta aí com mais de 1 bilhão de registros, e existem dados disponíveis para todos os grupos taxonômicos, para basicamente boa parte da Terra. Mas mesmo tendo esse volume tão grande de dados disponíveis, ainda assim, o nosso conhecimento é incompleto. A gente tem uma figura da biodiversidade altamente incompleta, cheia de lacunas de informação. Um dos fatores relacionados a isso é o sampling bias, ou viés de coleta, que está relacionado justamente à coleta em locais específicos, como próximos à beira de estradas, rodovias, rios, ou seja, próximo a locais onde você tem um acesso mais fácil para chegar nesses locais. E qual é o problema disso? Essas coletas, então, não são feitas ao acaso como deveriam ser feitas. E resulta nisso, um padrão de pontos de distribuição de pontos de coletas muito agregados, muito próximos a vias de acesso. Tá, mas qual é a consequência desse viés? A consequência são os déficits de conhecimento de biodiversidade. Eu vou citar apenas alguns, mas como exemplo, o déficit lineano, ou seja, nós não conhecemos todas as espécies que existem. Parte disso é causado justamente por a gente não ir até os locais que são menos amostrados, onde devem haver novas espécies a serem descobertas. Nesse exemplo aqui, nós temos justamente mostrando aqui com esse halo mais claro aqui, o número de espécies estimado para cada grupo, no halo mais escuro, o número de espécies conhecido para o grupo. E o que a gente pode ver é que para diferentes grupos você tem uma estimativa da falta de conhecimento sobre as espécies que existem, variando. No patamar máximo aqui, insetos com uma parcela muito pequena conhecida por enquanto das espécies. Um outro déficit de conhecimento é o déficit wallaceano, relacionado à falta de conhecimento sobre a distribuição das espécies. Vamos supor que essa área azul escura no mapa seja a área que a gente conhece como a área de distribuição de uma espécie. É possível que a área real dela seja muito maior. Então, a gente desconhece uma parte da distribuição da espécie. E a gente desconhece, entre vários fatores, por falta de coleta e viés de coleta. Por consequência, existe um outro déficit, que é o déficit hudsoniano, relacionado ao nosso conhecimento sobre o nicho da espécie, sobre as condições ambientais que a espécie ocupa. E nesse déficit, então vamos supor que essa esfera represente o que nós conhecemos sobre o nicho dessa espécie aqui, mas na verdade esse nicho é bem mais amplo, porque a gente desconhece ele, ou ao menos uma parte dele. Então, com todos esses problemas, o que nós podemos fazer? E será que esses problemas interferem diretamente em como nós medimos, como nós quantificamos a biodiversidade? Isso vai no cerne, no centro dos métodos biogeográficos. Todos os métodos biogeográficos têm como premissa de que o conhecimento sobre a biodiversidade é satisfatório. Uma das premissas comuns a praticamente todos os métodos é que a amostragem é suficiente para que você possa utilizar aquele método. O que, para amostras, por exemplo, em regiões tropicais, no geral, não vai ser verdade. Então, como que a gente pode contornar isso? Uma das alternativas para contornar isso por exemplo, como nós vemos nesse estudo aqui, esse estudo ele é focado em riqueza de espécies, padrões de riqueza de espécies em gradientes altitudinais. Só que um padrão muito interessante é que nós esperamos, pelo que nós sabemos de biogeografia, que a riqueza deve decrescer com o aumento da altitude. No entanto, quando nós olhamos para grupos específicos, grupos menores, famílias, ordens, esse padrão é muito diferente do esperado. Isso por quê? Porque esses grupos têm características ecológicas específicas, entre outras coisas. Mas quando olhamos padrões gerais, conseguimos ver o padrão geral esperado. Então, da mesma forma, nós podemos pensar que para podermos enxergar padrões gerais de biogeografia, dada as lacunas de coleta e o viés de coleta, talvez seja melhor utilizar dados de diversos grupos para que esses padrões possam emergir. Então, o que a gente fez em alguns de nossos estudos foi justamente isso: compilar uma grande quantidade de dados, no nosso caso, para o Brasil, para diferentes grupos taxonômicos, para que os problemas individuais de cada grupo pudessem ser minimizados ao se analisar o conjunto de dados como um todo, ou seja, ao se analisar o padrão geral. E coletamos, então tivemos aí mais de 25 mil espécies e uma amostragem filogenética também para as análises biogeográficas. Essa é a distribuição relativa de cada grupo na base de dados. E o que a gente vê é que os registros por grupo, eles são bastante díspares entre um grupo e outro. Aves, por exemplo, têm um número muito grande de registros, assim como algumas famílias de plantas, enquanto grupos de insetos e aracnídeos, no geral, têm menos registros observados. Por outro lado, o número de registros por espécie, dá para se ver aqui nas plantas, que você tem uma concentração de muitas plantas terem poucos registros por espécie. Artrópodes, né, invertebrados, você tem a maioria com um único registro por espécie, enquanto vertebrados você tem uma distribuição um pouco mais uniforme disso. Mas quando olhamos para o padrão geral, nós conseguimos então uma distribuição também mais uniforme. Então, o primeiro passo que a gente fez foi analisar qual o efeito desse viés, se ele existe, e como que ele interfere nesses padrões. Então, nós testamos aqui com simulações o padrão de coleta que seria ideal, no caso, essas linhas pretas aqui mostram isso, em relação à distância da estrada. E nós observamos que existe um viés muito forte. As coletas são feitas preferencialmente próximos a estradas e rios. E o que isso pode levar? Então, tem uma correlação muito forte no esforço amostral e as vias de acesso, e isso interfere diretamente em como nós medimos, por exemplo, a riqueza de espécies, o número de espécies. Existe uma correlação muito forte entre o número de espécies e quantas coletas eu faço em um determinado local. Tá, então como contornar esse problema, já que eu tenho amplas lacunas de coleta, eu tenho um viés muito forte que interfere nas análises? A saída que nós encontramos foi utilizar técnicas de interpolação, e essas técnicas vão, então, preencher essas lacunas, baseando-se nas evidências que estão disponíveis nos dados, técnicas de reamostragem, no caso de dados de riqueza, para que você pudesse ter uma contagem mais próxima da real do número de espécies. E com isso, nós conseguimos fazer, por exemplo, algumas análises de conservação um pouco mais realistas. Um dos dados interessantes que nós observamos para o Brasil foi para as unidades de conservação, unidades de proteção. Essas áreas protegidas, nós observamos que para a maioria delas, a gente tem muito pouca informação. Então, qualquer medida de conservação se torna muito complicada de ser realizada, porque falta estudos nessas áreas. E a gente precisa ter mais estudos nessas áreas. E um outro ponto que nós pensamos para a conservação é como identificar áreas mega diversas e prioritárias para a conservação. Precisamos de áreas com muitas espécies, muito endemismo, com muita história evolutiva, muito endemismo filogenético, e que ocupem as menores áreas possíveis, já que isso representa um bom parâmetro para a conservação. Construímos, então, um modelo que priorizava isso em cada região do Brasil, considerando padrões de betadiversidade, e com isso, nós identificamos essas áreas aqui em vermelho e amarelo como de alta prioridade para a conservação. Só que o que mais espanta nessa análise é que toda essa área cinza representa áreas pouco conhecidas, áreas na qual nós não temos conhecimento suficiente para tomar uma decisão em relação à conservação. Então, nessas áreas pode haver uma diversidade muito grande, uma alta prioridade para a conservação, mas ainda não temos evidência suficiente para conseguir sequer analisar isso. E isso também derivou numa análise de valoração de serviços ecossistêmicos na Amazônia, na qual nós também conseguimos expor essa ausência de informação, e numa tentativa, então, de conseguir reunir esses métodos que possam ser úteis para análise de biodiversidade, principalmente em meio a dados com viés de coleta e lacunas de coleta, surgiu o Biodinâmica. O Biodinâmica ele é um pacote de funções para o software Dinâmica Ego, na qual estão disponíveis atualmente mais de 60 funções de análise biogeográfica e macroecológica, e todas essas funções têm como opcional de saídas com meios de interpolação ou de predição por meio de variáveis preditoras ambientais. Então, com isso, é possível você minimizar os efeitos das lacunas de coleta e do viés amostral nas análises biogeográficas. E com isso a gente tem conseguido fazer análises, por exemplo, como essas, para identificar padrões biogeográficos em meio aos biomas brasileiros. Então, essa ferramenta, o Biodinâmica, ela possibilita de uma forma fácil, porque ela tem uma interface muito amigável, uma interface de fácil uso, que o usuário possa, então, minimizar esses efeitos do viés de coleta e das lacunas de coleta. O Biodinâmica ele utiliza internamente pacotes do R, do Python, na interface do Dinâmica Ego, para modelar ou analisar padrões biogeográficos. Muito obrigado. Thank you.
Hi, Ubirajara, and thank you very much for your presentation. It's really, really nice. So now we are going to start seeing the questions.
Okay.
Can you see the questions, Ubirajara?
No.
And stop sharing your screen. And now, maybe can you see them? No, not yet. Okay. Go and click in next to participants. Click in Q&A. Chat.
Okay.
Yes, and please read the questions because I cannot read in Portuguese. Sorry.
I don't see yet. Participants. Questions.
Did you find
okay. Yes.
Okay.
Uma das questões aqui, você usou em um exemplo a caatinga como exemplo de savana. O exemplo seria a caatinga do semiárido brasileiro? Sim, mas na verdade eu não me lembro de ter usado a caatinga como exemplo de savana. Uma outra questão aqui quando se fala em padrões biogeográficos nesse contexto, as questões estão se movendo. Nesse contexto, não são padrões de vicariância, e sim padrões macroecológicos. Há também, nesse caso, eu foquei aqui mais em padrões macroecológicos, mas também padrões como áreas de endemismo e padrões de diversificação também sofrem com esse problema, mas mesmo para análises, como análises de padrões de vicariância e dispersão, elas também podem ser afetadas por viés de coleta e lacunas de coleta. A grande questão é que, na maioria dos métodos que analisam, por exemplo, testam hipóteses de vicariância e dispersão, eles não consideram isso. E isso pode ser um problema, só que é um problema que, por enquanto, não está sendo analisado, mas pode interferir nos resultados, sim. More questions?
Yes.
Time is okay?
Yes. You have time to answer other questions. Don't worry. I will tell you when.
Uma pergunta aqui. Qual algoritmo biodinâmica usa? Máxima entropia? Quais variáveis? Então, depende. Para modelar distribuição de espécies, o biodinâmica usa o Maxent, né, o máxima entropia original do Maxent, assim como uma série de outros algoritmos, são 15 algoritmos para modelar a distribuição de espécies. Para a modelagem de padrões macroecológicos, como riqueza de espécies, beta-diversidade filogenética e outros padrões, o biodinâmica usa atualmente GLM, né, modelo linear generalizado, usa Random Forest e SVM. Em versões futuras, nós pretendemos implementar mais métodos para modelar esses outros padrões também, mas no momento são essas. Uma pergunta aqui. Opa, as perguntas estão passando meio rápido. Se quer entender o padrão de distribuição de um grupo... Se quer entender o padrão de distribuição de um grupo X, mas existem poucos dados sobre os grupos X e Y, possuem distribuição semelhante. Posso juntar esses dados e apresentar uma hipótese mais robusta? Sim, eu acho que essa seria uma possibilidade. No entanto, eu teria um pouco de cautela também, porque em alguns dos nossos trabalhos a gente demonstrou que os padrões de distribuição para diferentes grupos, no geral, têm baixa congruência. É claro, isso só diz respeito aos grupos que nós analisamos. Então, o ideal seria testar se esses padrões são similares ou não, para aí sim poder determinar se é adequado juntar esses grupos para testar alguma hipótese geral. Mais uma questão aqui. Gostaria de saber quais referências você recomendaria para estudar os déficits citados e se o biodinâmica funciona para atenuar déficits apenas no Brasil ou em outros países. Referências são muitas, tá? Tem vários artigos focados, já na última década, focados basicamente em déficits de conhecimento biológico e em como isso pode interferir no nosso conhecimento sobre biogeografia e macroecologia. O biodinâmica faz esse tipo de análise para qualquer lugar. A ideia dele é poder analisar o mundo inteiro. Então, ele funciona não só para o Brasil, o meu exemplo foi com dados do Brasil, mas ele funciona basicamente para qualquer lugar. Tem uma pergunta aqui sobre o uso...
Now you can answer a last question, okay?
Ok.
Thank you.
Sobre desenvolver a análise para o continente sul-americano como um todo. E sim, nós estamos ampliando o banco de dados atualmente, justamente para que possamos analisar o continente americano como um todo. Ok. I finished the last question.
Okay. Thank you very much. Thank you very much for your
time. Thank you very much.
Yes, wonderful presentation. And we have like 7 minutes of break before start with our Kingston speaker. That will be the person that will close in our symposium. Right, Salva?
Yeah. So please stay online, stay here, move some the body a bit, and stay ready for, in about 5 minutes, we'll start with the Dr. Carina Hoorn. Okay, we are very happy to have her here. And so please continue online, and we will start the talk in about 5 minutes.
Hola, Nicolás.
Hola, Karina, ¿cómo estás?
Bien, bien. Yo estaba confundida una hora. Pensé que era una hora antes, pero bien todo.
Ah, bueno, súper. No, me alegra mucho. No, créeme que yo también estaba confundido. Entonces, voy a compartir la pantalla y voy a compartir una pantalla para hacerte la introducción
Sí,
y
así
ya luego en
puedo
5.
poner la charla ya.
Sí, sí, sí.
O por lo menos la primera lámina. Ya no te puedo oír.
Primero, compartiré yo mi pantalla para hacerte la presentación y ya luego tú compartes tu pantalla. ¿Listo? En 5
Ujú.
minutos. Vale,
Ah, bien, sí.
perfecto.
Entonces me voy por un minuto.
Sí, sí, sí. Por ahora puedes, sí.
There is a lot of discussion here on our YouTube channel and here for the speakers. So we invite you to also continue these wonderful discussions on our poster sessions. You can talk with the people that give the posters, or we can talk with other people that are going that are moving around there. So please keep in touch using the Discord tool, and remember that make questions using the questions and answer option here, or you can also post your questions in the YouTube channel. So we try to move it here, and hopefully most of the questions can be answered online. Remember that after Carina talk, we continue our poster session. There is a lot of posters, so we will enlarge that session. So most of the people that per session and second session can have a lot of time to discuss. Okay, remember that it was expanded. The poster session was expanded. Okay. Nico, creo que podríamos empezar. Karina, ¿estás
Perfecto.
listos?
¿Sabes que en este mismo momento empiezan a ladrar mis perros?
Sí,
Pero no los escuchamos, no hay problema.
sí, sí,
No.
tranquila.
Ya los
Es-
escucharán seguro.
Eh,
¿Comparto mi pantalla ya?
no, not yet.
Ah, bueno.
Okay, we are very excited to start with our Keystone speaker. She is Carina Hoorn from the Institute for Biodiversity and Ecosystem Dynamics, University of Amsterdam. And her presentation title is Amazonia, Landscape and Species Evolution in Deep Time in Perspective. Like she have done like very important contributions to the knowledge of the geology and past climate of the Amazon and the ecosystems that have like a very important implications for the evolution of neotropical biodiversity, and you're very welcome to start, Karina. Thank you very much for being here.
Un segundo.
I want to just tell that Karina will be presenting in English, but she will be answering questions in both English and Spanish.
Ok, muchas gracias. Bueno, primero que todo, quiero darles un saludo en castellano. Me parece muy emocionante ser parte de este simposio y me parece una idea excelente esta forma de comunicar que surgió debido a la pandemia, pero pues parece una forma muy buena de poder reunirnos de forma virtual los científicos del Neotrópico. Entonces, haré mi presentación en inglés, entonces proseguiré en este momento. So, thank you very much for the kind introduction, and I'm very excited to be with you. I would like to talk about Amazonia, and in particular, about the possible origins of species diversity and distribution patterns. And I will do so by looking in a deep time perspective. So first of all, just to share with you the famous image by Bartlott et al. Showing the global biodiversity hotspots. And of course, I think all know the neotropical biodiversity stands out. But how was it formed? We know that the Amazon is particularly rich. You can see here a famous map by Ter Stegen showing the tree alpha diversity and the amount of species that he calculates exist there. So how and when did the species richness originate? That's a question that occupies many people. So I would like to take you through this journey of exploring the deep time perspective by first looking at the Andes and Amazonian coupled history. What is the role of the Andes in modifying Amazonia, and how has it affected environments? And then look at historical sediment archives that provide the evidence for this, both from land and at sea. And then also look at changes in flora from climate and landscape changes. I'm a palynologist, so I'm particularly favoring flora in this case. And yeah, and that's the content of my talk. So first of all, the picture of course, South America, and look at parts on how can we study this complex history. And as I said, events in the Andes affect Amazonia. And so this can be studied in 3 regions. First of all, of course, the Andes itself can tell us a lot about their uplift history, but also the sub-Andean and Amazonian sedimentary basins hold a rich record. And then finally, the submarine fan coastal region of Amazonia also has very rich sediment records. And all these pieces of information together can give us a broader perspective of what happened in this region over time. And I'll be mostly focusing on the Cenozoic, so from 66 million years onwards. I would like to start first highlighting the uplift as it is of the Andes as it's modeled by sediment chemistry. And I'm showing you here a map by Mauricio Bermudez on the right, the one that was used in our publication in 2010, which very vividly shows in the pink lighter colors the more recent uplift, and in the blue and greens, the more ancient uplift, and also a graph that illustrates this through time. And Mauricio has given me permission, Mauricio from Universidad de Sogamoso in Colombia, has given me permission to show the newest version of this map, which provides further details to this picture that already emerged from the 2010 figure, and which is also very interesting, of course, for understanding biogeographic regions in the in the whole of South America. But here we focus on the northern part. And you may be familiar with this map. Of course, Nicolas is very familiar with it because he made it. The map with the biogeographic regions for the central and northern Andes. And if we see his map, and he pointed out in his paper that there's a very important boundary between the central and northern Andes. And of course, if you look at the left to the map produced by Mauricio, you see very clearly also this differentiation in uplift ages, with the central Andes here in darker colors, and the pink, initiating here the northern Andes. So we think there's a lot of geological background to these biogeographic regions, and that there's a lot to be found out about that, and that a lot can be explained through the geological history, and especially the uplift. So not only in the Andes itself, you will have an effect on biogeography, and also the environment in Amazonia and the sediment composition, the bedrock, is affected by this uplift. And I think this is best shown by this system that was the Peibas system, which is a sedimentary system, and its geological name is Peibas Formation, Solimões Formation, that extends over 1 million square kilometers in Western Amazonia, and that was formed as a product of both uplift of the Andes, but also subsidence processes in the sub-Andean basins in Western Amazonia. And we think that affected very much the course of history and also of biogeography. And here again, this map that shows that part of this diversity coincides with this Andean substrate Peibas system. And just for reference, this polygon, this polygon represents the wettest area of Amazonia. So it's really the climate, but also we think that the geology and the Andean influence has played a role. But I will extend further on that in the course of the talk. And then I would like to add to this comparison of the influence of Andes and the substrate it creates in Amazonia, showing you this map by Hanna Tuomisto and her team, which appeared in the Humboldt special issue of last year, and which shows a composite showing the relation between soil variation, geology, and the fern and lycophyte diversity. And you see also these different colors for central Western Amazonia in relation, compared with Eastern Amazonia, which is the crystalline, craton, shield areas, which are very different lithology and much older as well. Of course, everybody will know this image of the meeting of the waters, and I'm using it here to show one more time this intricate relation between Andes and Amazonia, in this case the Andean and the Amazonian rivers, with the black and the crystalline rivers born in Amazonia being more nutrient depleted, and the white water rivers being born in the Andes and being highly fertile and generating fertile soils in the floodplains and so on. So how can we piece together this history? Where do we get the information from Amazonia to get to a reconstruction? So I said there's archives at land and at sea, and a lot of information comes from drill cores that can be because of scientific purpose, because of water drilling, water resources, or hydrocarbon. And so on the left you see example of drill cores that reach almost 1,000 meters, which were drilled by Petrobras. That's a very good information source for deep time history, but also along the outcrops, along the rivers, there are outcrops with pieces of history of the Amazon, such as here you can see this Solimões Peibas formation. This is near Leticia in Colombia. And here you see a cross section like, what can we tell from the subsurface? And these are the basins, the areas that hold the sediment records. And here for reference, you see the Andes on the left, and on the far right, the Foz do Amazonas. So it's a profile across the continent. Well, my history in Amazonia goes back to the late 80s when I went to the Araraquara research station along the Caquetá River. And here you see the Paleozoic sandstone table mountains that are just situated there at Araraquara. And it's along the Caquetá River that a lot of these outcrops can be found. Of course, it's always a search because it's a rainforest area after all. But nevertheless, together with information from Brazilian drill cores and outcrops along rivers such as the Caquetá, but also, of course, the other tributaries of the Amazon, a lot of information can be obtained. So I'll show you some examples from the Caquetá River. So this is, for instance, what we found out are early Miocene sediments. So that's about 20 million years ago. We can use pollen that can be age, can be an age indicator, markers. And so, for instance, we use the pollen, the reproductive material from plants, to not only get the age, but also to establish, for instance, past vegetation composition. And we know from that the early Miocene had features of a tropical rainforest, although it was somewhat different still. Just to give you further idea, so we can also look at the source of sediments by looking at the sediment chemistry, and we can tell whether the source were Andean or cratonic. And from that and other information, we came to this model that at least in the early Miocene, we still had a drainage division in central Amazonia, much like also was the case in the earlier Cenozoic and possibly also the Cretaceous, with an eastern Amazonia that had a proto-Amazon sourced in the Craton, and another drainage system in western Amazonia that was mostly directed towards the north in the Caribbean. And so that this fluvial system was a Craton-born river system that was directed in the opposite direction than now most of the tributaries which are leading to the Amazon and to the east. Just to jump further throughout this history, so large part of the Western Amazonian surfaces are relicts of the Peibas system, this Peibas solimois formation. It's a clay unit with wealth of invertebrates like mollusks, but also ostracods, and also wealth of fossils that has been studied by many paleontologists, and wonderful information has come out of that. So I just want to give you a little flavor of that by showing you some examples. For instance, the mollusks were studied by Frank Wesseling from Naturalis, but the large fossil remains, for instance, Rodolfo Salas Gismondi from Lima, Peru, he has documented these, Douglas Riff from Brazil, and there's other researchers working on this wealth of fossils. And for instance, from the reptiles, it has been shown that they were very diverse in this Peibas system, and also fed on the invertebrates that existed in that system. It was a very unique environment that is thought to be a long-lived lake system, or with also contribution from fluvial systems. But this system, of course, disappeared at some point, and then was recolonized by other flora and fauna. Just a little bit more on these reptiles. And then our model proposes that around between 10 and 9 million years, well, this Peibas system disappeared, and we come to that idea that then it migrated into this transcontinental Amazon river system, and that a lot of that information comes from actually the coastal region of the Amazon submarine fan, which is a very good record, a continuous record of both coastal history, but also the influence of the Andes on the coastal system. just a photo on the Acre region, and one of the fossil outcrops. A lot of the reptile fossils and many other types have been found in Acre and worked out by the Acre paleontologist. So yeah, so now a little bit more about this sediment record at sea, at the submarine fen. And so the evidence that that is available is mostly sediments that have been drilled by, for instance, oil company like Petrobras, or in collaboration with other oil companies. And so they have managed to do a deep drilling, recovering about more than 5 kilometers of sediment. And that sediment tells us about the history from land, but also from what happened at sea. And here you see in green this algal blooming that is related to the sediment that the Amazon brings in. And of course, we mentioned at the beginning that the Amazon brings in nutrient-rich waters that determine what happens on the floodplains, the fertility of the floodplains, and the richness of the biota. And this is also has an effect at sea. So the plume brings in nutrients which compost the ocean and which foment primary productivity. And this is something you can also follow in the fossil record, which is interesting if you want to understand biotic evolution at sea in the equatorial margin of Brazil, for instance. You see, for instance, also here how the Amazon River, of course, affects the coastal region towards the Caribbean, and in blue here the freshwater plume that of course affects also the oceanic water. So what can we tell from the sedimentary records in terms of how species change in composition in relation to climate and geological changes? So a little bit about this marine record from the Amazon fen. So a couple of years ago, quite a few years ago now, we had the chance to collaborate with geologists from Petrobras and sample this Amazon submarine record. We had two different studies, one on studying the pollen composition to see if we could reconstruct history of the hinterland, which vegetation groups were providing pollen to the sediments that landed in the deep ocean. But also we looked at what was the algal composition of the dinoflagellate cyst. And Emmy Lammert's map published this paper in 2018, where the results were shown that when we look at the transition from the situation before the Amazon reached the Atlantic and after, we see that the sediment change that was documented by Brazilian geochemists, and that is seen between 9 and 9.4 million years, we saw also other changes. We saw also biological changes. So for instance, the composition of the algae changed from those that feed on nutrient poor, feed in a nutrient poor situation to a composition where we find algae that feed on nutrient rich waters. And so that was a very interesting change, and that coincides, of course, with the change in input from mostly land derived from initially a situation where you have hardly any land input to a vast increase of fluvial input, which is the arrival of the Amazon River. And this is also followed by changes in foraminifera that characterize the bottom conditions of this coastal system. So that's what gives us a clue on how the productivity in the ocean changes. And just to give you an idea, this is what they look like if you magnify them vastly, the organic walled ones and the calcareous walled ones. And then we also looked at, of course, at pollen to see what could we tell, and this is on the right side of the figure. I'm not going to go into the details, but just want to highlight that after the arrival of the Amazon River is registered at sea, we see an increase of tropical mountain taxa, open Andean taxa, and also we see a rise of grasses. And this left us very eager to understand, well, what can these records and also the land records tell us, for instance, about the evolution of important groups like the grasses. So if you then look at what is the sediment that is brought in by the river, of course, it's a very, very big drainage system. So the sediment source and also the source of pollen, of grass in this case, that was what were focusing on. There's a number of different biomes. For instance, it could be from the high Andes, if there was a high Andes already. It could be from floating meadows in the lowlands, or it could be even from the Cerrado or from the lowland forest. So just not so long ago, we thought further about this and compared also with data from land, from this PEBA system that I introduced to you earlier on. And Jaramillo et al., actually this should be 2017, not 2018, published a paper in which they show this record. It is very rich in grasses, and this is in the early to middle Miocene, at the time when the PEBA system was still there, this wetland. And we think that in comparison with Eastern Amazonia, where you see very few grasses arriving in the Amazon fen, that this suggests that actually the PEBA system might have had already typical floodplain grasses growing there, and possibly also contribution from the Andean slope. And then we think that the upper section of the Amazon fen shows us that there's an increase in grasses that is likely related to the Andean slopes and also already to the high Andes. So that's what this record taught us about, for instance, grass development in the Amazon basin, Amazon drainage basin. So what else can we find out from the terrestrial records, from the PEBA wetland and from the drill cores on land? One of the aspects that really interested us and that has been coming out over the years from different records is the marine influence in Amazonia. And of course, marine influence would have left also maybe relict taxa, that coastal taxa, that also could affect the modern biodiversity, or at least to understand how to understand the modern diversity patterns could also be seen in the context of these marine influences. In this map you see, this is a map that was compiled by Melanie Boonstra in 2015, and you see here all the dots are representing locations where marine influence was observed. And so you see that it seems like a pathway from the Caribbean deep into the Amazon. There's never evidence for true marine conditions or really coastal. It's always very extreme, to the extreme of really almost fresh water. So very oligohaline to fresh water. So it's very, very subtle influence really. But it is reflected in, for instance, in the invertebrates, fossil invertebrates, fossil organic linings of foraminifera. And if you think of it, also the modern taxa that still populate the rivers, you would think they have a relation to these ancestral marine incursions. This map is a little bit of an exaggeration, showing possible scenarios. We think, as I said, it's more like estuarine conditions, very, very faint influence of marine conditions, but still enough to bring in taxa that are from the coastal regions. Just recently, we looked at the palynological assemblages that hold mangroves, and there are certain sites, especially in Colombia and Amazonia, in the Apaporis region, where the fossil record has very large amounts, up to 80% of mangroves, which are really typical coastal taxa, and you normally don't find them far out of their place where they grow. And so an analysis by Chumbata et al. Suggests that actually these mangroves, the Rhizophyra, had a whole assemblage of other taxa associated. And this plays into the hypothesis that was published by Bernal et al. Last year, also in the special issue in the honor of von Humboldt, which suggested that a number of estuarine taxa are presently occurring as relics in Amazonia. And so that's why we looked, what does the fossil assemblage hold? Is it are there some resemblances, which what Bernal suggests for the relic taxa. And we did find at least for this whole assemblage of about 14 taxa that were associated of the mangroves, we found at least one that we could identify and relate to a modern taxon, which is Humilia balsamifera, which is known from the Restinga environment, which is the coastal sandbars in Suriname and Brazil. So there is not just the mangroves, well, not just, they are a very important indicator, but it looks like they had a whole assemblage of other taxa that they brought into the heart of Amazonia. And this is something that is interesting to explore further to understand what part of modern diversity can be related to these ancient marine incursions. And this is here a little map to give you an idea of what this pathway of the marine incursions looked like. Then also would like to highlight that when we look at the marine records, for instance, when we look at the sedimentary record from the Pebas system, from these fluvial, the relics of these old fluvial systems, we also find large abundances of palms. Palms are occur in large abundances in the fossil record. And so we focus, for instance, on the Maritime palms, which are endemic in Amazonia. And we have been looking just recently, this is a paper that's still in preparation, looking at the history of the Maritime palms and see that, that at least some of the ancestral Maritime, such as Grimsdalia, was a palm type that was very prominent in this Pebas system, and that disappeared after that this system also vanished. And we suspect its history is closely related to that, and that also on the larger scale for the Mauritia and Mauritiella, this landscape changes may have played a role, but in a different way. For the Mauritia and Mauritiella, it may have been very favorable. These changes in environment because it's endemic here. And if we compare with the larger fossil record from around the globe, and I don't have the map right here, but they were pantropical and they existed all over until from South America to Africa and reaching even into India. And so they disappeared, their global record dwindled, but actually the only place where they really stayed prominently was in South America, where they became an endemic. And we believe that this system, this development at the foot of the Andes and the conditions that were created, they were actually suitable and favored their development in Amazonia. Well, I also would like to highlight another aspect, and I think that has been proposed by maybe by Alexander Antonelli for the first time in 2009, or at least illustrated very well, which is this pathway from Amazon to Andes. So I've been looking at developments within Amazonia, marine influences from Andes to Amazon, but you can also look at species in the context of movement from Amazon into the Andes. And an example for this could be, for instance, the history of the Iriartei palms, which has been documented by Bacon et al. And so this is a taxon that is actually originally mostly known from the fossil record in the lowlands. And so I've highlighted here three areas where we think former ancestral Iriartei have been documented, for instance, such as in the Eocene, in what was called the Barca concession, but also in Cretaceous sediments. And it should be noted that in the Cretaceous, the Eastern Cordillera wasn't there, so it was a lowland at the time. And then, for instance, in the Miocene of the Pebas sediments, we find these Iriartei type pollen. However, nowadays they're much more common on the Andean slopes. And just for illustration, this is what they look like. And so this seems to be a group that has been taking advantage of these developing slopes and migrating from lowland into this Andes following the geological developments. And then one final example for this is the example of the Malvaceae type pollen. We followed two groups of pollen that we know are common from the Miocene in Amazonia. One group belongs in South America, as a Gondwanan type, and another type, which is this one that you see here, the black and white picture, is an Abutilon type, which is sort of immigrated into South America from North America. And here you see the phylogeny that has been compiled by Laura Loga-Morcino. And as a green bar, I've put here the moment of the uplift of the Andes. And this taxon is initially only known from the lowlands. And then it seems that when you compare the distribution maps, that especially in the, Quaternary, Pliocene Quaternary, it starts colonizing the Andes and is dominant there. So I think that's another example of where taxa have taken advantage of the developing mountain system and taken to the slopes. So those were some examples that I wanted to share with you to illustrate that the geological and biological history of from Andes to Amazon is an integrated process, and that it's to improve biogeographic models. It's important to have accurate and up-to-date geological data into these models, and that this could go a long way also in terms of choosing conservation areas to preserve global biodiversity. Just for some, suggest some further reading if you like. There's these two books out there that I co-edited with colleagues, with Alessandro Perigot and Alexandre Antonelli, the book on mountains, and the Amazonia book, which was co-edited with Frank Wesseling. And then I also would like to call your attention on a new issue that we are preparing, and we hope there will be many people interested in contributing to it, which is issue in the journal Global and Planetary Change of Elsevier. And they have given us the opportunity to call for this issue that explores the impact of the uplift of the Andes on evolution, climate, and landscape modification. So I hope this is a good moment to reach out and to entice you to work in the context of linking geology and biology to understand mountain diversity and also Amazonian diversity. And so, you can find this announced at the site of the journal as well. So that's what I wanted to tell you, and it was wonderful to share this time with you. Thank you very much.
Okay, thank you, Carina, very much for your presentation, also for accept our invitations. Like, we are very happy with your wonderful presentation. So here in the Q&A questions, like you can see some questions, like for now.
Okay, where do they start?
Like, I can read to you the questions if you want.
Okay, yeah.
Is there a current ecosystem that is similar to Peba's system in the Miocene?
Yeah, I always wonder about that as well. I think maybe the Pantanal, I see Camila Ribas there as well. What does Camila think? I think maybe the Pantanal.
I've thought about the Chaco.
Okay.
Yeah.
Chaco as a possible remain of the Peba system, but yeah, Chaco and Pantanal.
Yeah. Yeah.
Okay.
Thank you, Camila.
I also have a question.
I sometimes, yeah, no, I was going to say, sometimes I wonder about the Varzeas and comparing the Varzea sedimentation with the Peba sedimentation as well. I think there will be some resemblances, but no, it is a very unique system that I think also, like, for instance, Carlos Jaramillo says it has no analog, and I think also in the past Frank Wesseling and Hubert Vonhoff, they've also pondered about it. I think it's a very unique system that has estuarine characters that doesn't exist at present. No.
And also the Várzea system is very dependent on sediment input from the Andes. So it depends also on the Andean dynamics to know if the Pebas was a big Várzea or not, right?
Yeah, the one thing that has become clear from sediment analysis is that there is a cyclicity. So it's a long time ago, I think beginning of 2000, Hubert Vonhoff did this sampling, and there is about 7 or 8 cycles in an outcrop of about 30 meters. And so we are now comparing the palynology, and we see actually also sort of palynocycles, and with the invertebrates it's the same. So there are probably parts of the Pebas that are resembling a Várzea, but overall, so because of course there was already this fluvial input, but was there a flood pulse like it's now?
I have a personal question also that I want to take the opportunity to ask, and is what do you think that are the factors that cause that the Orinoco region in Venezuela and Colombia are not forests but are savannas ecosystems?
Oh, there you ask a different question. Yeah, I'm not sure I have the answer to that. I think it must be because of the, I don't know if it has to do with the displacement that because of the northwards movement of the continent as a whole, it has moved into a different climate zone over time, but the movement hasn't been large. So, yeah.
Okay. Okay.
Can I make a question as well?
Yeah.
Yes.
So Carina, you weren't here before. I know you were in the other meeting.
Yeah.
In which I should be as well, but I wasn't. But I was talking, as always, you probably saw a similar talk in the meeting last year in Ecuador about bird diversification. That seems to be quite more recent than the time frame you were discussing now.
Yeah.
In your talk. Mostly happening between 5 and mostly after 3 million years ago.
Yeah.
So we keep trying to discuss what was happening in Amazonia during that time frame. I remember in the 2010 review, you have that the review is a longer time and the establishment of the drainage about the end of the Miocene. And I would like to ask, how do you see it now after 10 years? What do you think happened after the end of the transcontinental establishment? And how dynamic was the Quaternary geomorphologically speaking?
It's good that we discuss this. Yeah, it's. I mean, this is something I tend to go in more when I'm talking only about the Amazon River development. And so I think through also with this work that we did on the Amazon Fen, and so thought much more about this. And yeah, I think one thing that's very important in that sense is the change of this transcontinental river system. So one thing is the start, which is the first sign of its arrival. And that's why the definition of Amazon River needs to be considered. So the start would be the arrival of Andean sediments, but that's not the system, how it evolves. It changes very much through time. And I think a lot of that is also already why this grass curve comes in, is because there's a vast increase in sediment because also of the climate changes that are taking place in the basin. So I think that's something different from 2010. So once I will learn more about the history of the river, but also that, yeah, it's very important to look into deep time, but at the same time, the more recent processes, like what you are pointing at, the 5 million years, they have also reshaped entirely the landscape. So the fluvial system has incised and flooded, and so the climate has acted. So yeah, a lot happened. So you need a deep time perspective to understand the ancestry of your forest and of your organism, but at the same time, the landscape has modified an awful lot. And I think it's not well enough understood, really. There's no long sediment records. Of course, the Amazon Fen is a good record, but it's like a bin for all the sediments. And it's very difficult to, maybe if the Trans-Amazon drilling, this drilling project that is for academic purposes, if that will happen, then maybe this kind of records will be available. And also if they do the planned cruises for obtaining more coastal sediment, then you may have a much better insight, even though it's like an overall view, maybe coming apparent. But this is one thing I've learned in the last 10 years is, yeah, the landscape modification has been big. So one thing is the ancestry, but the other thing is the modifications afterwards. And I think you have to see both perspectives to piece the history together. So yeah.
Okay, and the next question is what is the importance of the Purus and Vaupés arcs in the Amazonian formation?
Yeah, those arcs. Well, the Vaupés arc is very important because it divides the Orinoco basin and the Amazon drainage basin. And these basins, and I didn't actually mention it, but yeah, they were connected until the Vaupés arc was formed. These basins were connected, and you had a pan-Amazonia that extended up to Maracaibo. And so the Vaupés arc is, for that reason, already very important because it's together with other geological processes, it confines the Amazon River to its present drainage basin, I think. And the Purus arc, well, I think it's a bit difficult to gauge exactly what its meaning is, but we know that when the Andean sediments reach the ocean, there has been a change in the geometry of the basins. I'm not sure if it's really the arc itself. It's a marker point for the basin division, but it's more likely that it's the combined uplift process of the Andes and the changes in subsidence because of mantle processes that has changed the sediment accumulation balance. And so it moved from sedimentation in the sub-Andean zone to sedimentation that migrated to the coastal region. So I'm not sure the Purus is more like the marker point, but it might not be something that actively changed. It might be on either side of the Purus where things change, in particular in the western part of Amazonia.
Okay.
So it's like tipping the balance. Where the Purus arc is like sort of the marker point with the flag, like, okay, from here, suddenly now it's no longer a barrier, but it's now a continuous.
Okay. And the next question is, the integrations, dear Carina, the integration school work has corridors for savannas?
Sorry, the corridors for savannas?
The integration school work has corridors for savannas?
Not sure I understand. Maybe in the marine incursions he means, or he or she means, maybe.
Yes. I should not understand. Let's move to the next questions. Thanks, Carina, for the great talk. I thought very interesting the mangroves and Restinga elements inside Amazon. Regarding Restinga, it is relative recent community. Do you know the age of the pollen presented?
Yeah, so this is in the Miocene. It's the middle to late Miocene. So I don't know how young they have been dated, but for us it was a real surprise because we didn't know where to fit this pollen type. Well, that it was a humidia, and it became very clear that it had to be a balsamifera type. It wouldn't be exactly the same because it's a Miocene pollen type we're talking about, but it looks like it's an ancestor of that one. So I don't know what the phylogeny of the balsamifera tells us, but yeah.
Mm-hmm. Okay.
I would say there's a lot more to explore on these mangrove communities and the relics on land. So this is where I recommend Bernal et al.
The next question is there a register for extinctions, fossils related to the periods of marine incursions?
Extinction event? Is that what's meant?
Yes.
I don't know. It's a good point. You would expect that. And I think that's not something that has been looked into properly. It might, I mean, as I said, they are very clearly a lot of, it wasn't a major extinction event. So that probably also supports the idea that it was like very very diluted saline water, so hardly saline at all. So that's maybe why it didn't affect taxa in a very big way. I would say the biggest extinction event, which still is underdocumented, is actually that from the invertebrates, because the mollusks and the ostracods, they are definitely the diversity of mollusk and ostracod that existed in the Pebas system totally was decimated. And so now what I understand from those who study this is that it's like a fraction of the diversity that existed, in the same way as the reptiles seem to be much less diverse than they were in the Pebas system. So in terms of extinction event, I think the draining, the emptying of the Pebas system was an extinction event, and the marine incursions are, I think they're more like a way of transporting organisms into the land environment where they possibly could adapt, because there was not a very strong gradient in salinity. So, but that's, I mean, I speculate about that. It's something that would be worth investigating further.
Okay, now like there is no a question, it's just like a comment. There is a like about the Orinoco's issue and savannas. I think that there is a relationship with the soils.
Well, we've learned from Beto Quesada in the science panel of the Amazon that the soils are very directly related to the bedrock, and we had some inkling on that in 2010 by looking at the general soil map. But it's very nice work by Beto Quesada from 2012, and where it's very well documented what the contrast in soils are between the crystalline rocks in the Craton in Eastern Amazonia and Western Amazonia, and how everything is different really. The soil profiles, the rate of growth, even the groundwater seems to be determined by the bedrock properties. So yeah, so soils definitely will be different in the Orinocan region because of that.
Okay, and the last question is, the changes in the course of Araguaia to Catins Basin can also be detected in the sediments of the Amazon fan?
I think, well, this would be a question for the sediment chemist, but we do know that there was there are some fluctuations when you look at the composition that are of local origin. So you get throughout this Amazon fan record, there seems to be input from the nearby, the proximal region. So yeah, there's definitely things happening there as well. It's not like stable and only coming from the Andes. No, it's not like that. There's definitely fluctuation. So yeah, the answer would be yes, but it's something that I know from the sediment chemists who work with us.
Okay, and okay, we finished. Like, Carina, thank you very much for your wonderful presentation. Also, we want to thank all the speakers, Camila, yes, Camila, Jonathan, Angie, Ubirajara, like all of you, like, thank you very much for all your time and valuable effort. I don't know if Salva want to say something.
No, I also want to thank all of you for your time and your great talks. We are very happy because we have a lot of participation in the questions, and also a lot of people chatting that they send you a lot of good, they are very happy with talks. So it's up to you that you make all very nice work, and we are very happy to have you here. And now I invite you all the people to continue this discussion on our Discord server. We will have a poster session, so you can talk with the poster presentators, or you can talk with anyone that you find going on there. Again, thank you thank you very much for all the Grok talks. I'm very happy to have you here. We are very happy to have you here.
Also, I want to tell to all the speakers that just like an interesting information is that during all the symposium, we have around 250 in audience in the Zoom platform, and we have like around 150 in the YouTube channel. So at the end, like around 400 people watched this symposium. So it was like a really, really, really good. And thank you very much.
Well, thanks a lot for the initiative. Really nice.
Okay.
Yes,
okay.
thanks a lot. It was
Yeah,
great to
thank
participate.
you.
Okay, bye to everyone.
Bye.
Thank you.
Bye.
Bye.
Bueno, chao, que estén bien. Muchas gracias.
Chao, que estén bien.
Muchísimas gracias a todos.
Vale,
Gracias.
Gracias. Hasta luego. Cuídense.
igualmente.
Listo, yo no sé.
Hasta luego, Salva. Cuídate.